a natural history study will allow you to target the most important aspects of the disease which you try to treat. And this is important. This goes all the way down to designing the drugs, because it tells you which cells, for example, in the brain you want to target, so you would have the effect you're aiming for. And you have to set relevant outcomes for that. So a well-executed natural history study prepares a group of patients, in our case, a group of CTNNB1 patients for clinical trials. I'll give you two examples of how this can help. And this is the first example is the Duchenne muscular dystrophy patients. It's a disease that affects mostly boys. These boys will develop quite normally in the first years of life, but then they will develop muscular problems for which they will lose their ambulation and so forth. And in this particular study, they looked when, for example, the patients would start to have cardiomyopathy, which is the green line, and you can see that more and more patients with age would develop that problem. Also, going in line with that is the ventilatory problems, for example, the orange lines, and you can see that these problems are much more frequent and occur, in a bigger percentage of patients. So if you have an understanding of the disease in this sense, you can then look at how the treatment is helping these patients. And here, if you concentrate on the orange line here, which is the natural history study of patients. I mean, these are patients who have never been treated. You can see that, and if you look at what age they lose ambulation, so when they stop walking and need a wheelchair assistance, you can see that the orange line is falling down, and about 50% of patients will lose their ambulation at about 12 years. And if you now compare this to the blue line, which is the cohort of patients who have been treated with steroids, you can see that this is now significantly different to the cohort of patients who have never been treated. So, for example, in this study, they were able to show that by giving patients steroids, they were able to prolong ambulation for about 3 years. And you can go even a bit further. This is an example of spinal muscular atrophy. Here, the patients are losing their motor neurons and they become increasingly weak. Also, so if you look at the natural history studies, which are the orange and the gray lines at the bottom, and you start treating these patients when they already have developed symptoms, you see that these patients would increase in their motor capabilities. But you can go a step further in asking the relevant questions. How does it help if you start treating these patients really early? And you can see that if you start treating these patients in the pre-symptomatic phase, many of these patients will increase much more than if they were treated symptomatically. So, and if you're able then to compare all this to the natural history study and show that this medicine is really working. And this is incredibly important when you have, even if we are lucky enough to, at some point, we'll be able to have a therapy, we need to know whether this therapy is safe and if it's efficient. So we already, I mean, many of you have participated in the genotype phenotype study, and I'm happy to announce that we are just about to finish all the analysis, and the paper is almost ready to be submitted. And but this was a snapshot in time. So, for example, if you look at the lower part of the picture, when we were looking at walking, we were able to see that 50% of CTNNB1 patients that participated in that study were able to walk eventually. But as you can see, many of these patients would start walking close to 15 months, so quite late. So we understand now that our patients have a problem in walking development. And we've looked at other things here as well, when they were able to crawl, when they were able to babble and so forth. And we can say something about the disease. But as I said, this was a snapshot in time, and we need to understand how this develops over time. So besides this being a scientific congress, it's also a beginning of an international natural history study. And, as we are speaking, there's patients being evaluated on different domains around this hotel, but we'll also take them around to our hospital, but also to the ophthalmological clinic to be properly examined. We have had an ethics committee approval from Slovenia for the protocol of the study, and we will adapt it if necessary. If we find out that what we're doing now is maybe not the most appropriate thing to how to examine a child, we'll adopt it to best suit our patients. We aim to be able to follow the patients for at least 5 years, but there's always a chance that we can prolong it if everyone is agreeing on participating even further. And what we are also trying to do is to make it kind of more easily available for the patient. So we do appreciate that you came for very far around, as far as possible, really around the globe. And I know that this is a very big burden for to travel with kids that are not so keen on traveling. And we might be able with time to kind of have multiple sites which will be able to examine our children with the same method, collecting the data in the same database, and we'll still be able to get the relevant data that we are aiming for. So we also, we will anonymize the data, and we will definitely analyze them and publish them. So it's available to the whole community of CTNNB1 researchers around the world. And I think this is how science should be done. So there's a couple of aims that we kind of placed in the protocol about the dragonfly study. We want to see what the signs and symptoms of the disease are. We know that although the same gene is affected in all of our kids, the different mutations can be related to different phenotypes. Some are more severe, some are less severe, and we want to understand that even better. We want to understand what tools do we need to evaluate our children, because we need to have such tools to be able to say something about a particular child and how the child is going to develop. And we also try to understand what the impact of the disease is on patients and their families, because obviously we will try to aim for a treatment that suits mostly, the most important kind of relevant issues for the patients and families. We will try to collect some biomarkers, and here I would like to underline again that all this is completely voluntary. If your child is not able or doesn't want to, or you have other reservations about any kind of examination, please let us know. Nothing is obligatory here. We also try to have a snapshot of whether the parents are interested in participating in future clinical trials, because we are already preparing a clinical trial in the case that we'll have a successful treatment in the following years. And I strongly hope so and believe so. But of course, we have to follow the rigorous scientific and legislative procedures to be sure from all possible perspectives. We also need to, the good reason about to have such a natural history study is also to connect. And I think we are doing a great job with connecting with all of you and all of us. So as I said, there have been a lot of families coming from all around the world. It's been said before, 82 patients and families from 19 countries, which is incredible. I'm still overwhelmed with the fact that we managed to get all this this amount of patients to Ljubljana, and I'm incredibly grateful to all of you for injecting all these efforts to make this happen. Participation is completely voluntary, and as I said, you can always say no to any of the to the examinations that are being performed. We have, I don't want to burden you too much with everything that we'll be doing here, because you will learn this on the fly, but we have generally kind of for the sake of examinations, divided our patients into 3 groups of those who are less than 4 years, those who are more than 4 years and are non-ambulant, and those who are more than 4 years and they're ambulant still, because of course not all examinations can be performed on all kids. We will be collecting some existing data. For example, those patients who were able to get Bayley scales of infant and toddler development or Wechsler intelligence scales, we asked them to bring them with us. Also, the EEG recordings, if they were performed in the local environment, we kindly asked the parents to bring them with them. And also the MRI scans, we don't have the possibility of doing MRI scans here, but this is also a very important thing for understanding of what this gene mutation does to the brain development. We know a bit more now. We know that how neurons kind of connect among each other in the brain is kind of disturbed in the CTNNB1 patients, and we want to understand this much better. And for that reason, MRI scans are really valuable. So we'll have each patient will be clinically examined, and we'll take some demographical data. We'll ask you about past medical history, some comorbidities, and then a general medical and neurological examination is going to be performed. We'll also monitor the motor milestones, and there's some other fine details. We're going to look, for example, for dystonia in your patients, in our patients, and you will. It will last about an hour. Looking at cognitive function, we look at Bailey's scales of infant development and Wechsler preschool primary scale of intelligence. And looking at language and communication, we also will use these two scales, but also look at other assessments such as adaptive behavior assessment system and Vineland adaptive behavior scale. When looking at adaptive skills and behavior, we'll also look for autism, and we have different questionnaires for detecting problems in that field. And we'll also ask about the quality of life measures related to your family. As I said, MRI and EEGs are going to be collected if possible. And for the EEGs, we also had the opportunity to record some in our hospital. And we'll also do the optical coherence tomography, which is a retinal scan. And this will be also an important measure to evaluate because we know that eye problems can be part of the syndrome. For a selected group of patients, we'll also use a wearable device. Laurent is leading the field in this part because this is a device that you can place on the ankles of the patients, and then using very sophisticated computer analysis, you can actually see how the patient moves, and maybe this is an additional tool like a marker that will be able to show us whether, how the patient develops, but also whether if you treat patients, whether the patient's movement is actually having some sort of qualitative changes that can be appreciated and will show us whether, the treatment is effective or not. And this is, for example, a device that has been approved, for example, for DMD patients. And it is one of the important outcome measures in any clinical trial related to that group of patients. We'll also make photographs of children because we would like to see whether we can use, again, artificial intelligence in recognizing these patients. And Phase2 Gene is it's an academic kind of collaboration, and they have been very successful in putting a lot of logic behind, facial recognition, connected that with the genetic problem. And you probably know that the CTNNB1 gene is a very common gene which you can find in cerebral palsy patients, because we now know that about a third of cerebral palsy patients actually is not cerebral palsy as we know it. It's not a problem of birth, but it's actually genetically caused. And CTNNB1 gene has been found as one of the most common genes. So if you had such a tool, for easy recognition, then you could use it in clinical practice. And even if you're not very familiarized with the CTNNB1 syndrome, and the program will tell you this might be a child with the CTNNB1 children, which is very important for early detection of these patients. As you can see, as you were able to see on the previous slides, early treatment is also usually related to a much better outcome. For those who will be willing to do so, we will draw some blood. It's going to be 8 milliliters of blood. We'll try to analyze it for DNA methylation, which is a method of looking at how DNA changes over time, especially if a certain intervention is being introduced. We'll look for RNA expression, and we'll look for various biomarkers. For example, neurofilaments is a biomarker that have been used with success in the SMA field, and it's a marker of neuronal degeneration. So if you would have a treatment and you would see a fall in the levels of neurofilaments, you might be able to say that your treatment is effective. So with that, I'd like to conclude and say that a natural history study is truly a team effort, and it requires massive collaboration. I think us sitting here is a proof that this is possible in the CTNNB1 field. And I thank you all, to all the patients, all the families, and all the healthcare professionals, which we heard are more than 30 volunteering to do the examinations here. And all the volunteers, there's over 100 of them helping in managing the whole process. And I think this is a very important and crucial step to the next phase, which is a clinical trial of a potential disease-modifying treatment. With that, I would like to thank you.
Dear Damian, thank you very much. I'm still happy to see the dragonfly as the sign.
I will come.
There is a DNA strand in the middle. You know, the icon has a DNA in the middle.
That's true. That's true. That's
so
important part of
that's something nice.
Story.
And I see a connection with Ljubljana, you know, being the dragon city, you know.
Yeah.
So for whomever, again, plugging Slovenia.
Sorry?
Plugging Slovenia as a tourist destination.
Yeah, we're selling Slovenia very well so far. So
I will continue to do so.
Okay, great.
Okay. So before continuing, thank you, Damian. Thank you very much also to the translators. I know it's a very hard job, and thank you very much. And now we shall continue with Dr. Laurent Servais from Oxford and Melissa Menard. And they will tell us about the clinical relevant endpoints. Just as a side note, some of the scientific information might be scaring a few of us or you or whoever. Don't be scared. It's actually not as hard as it seems if you get the right teachers, if you get the right people to tell you about the most pertinent questions. It's also important to understand that hope has to be balanced. So it's not just about hyping everybody here, because if it doesn't succeed, the desperation and the disappointment will be too hard. So we are entering a field that is new, and there are risks, and nobody's telling you a story where you should just trust and that's it. No, ask questions. The questions that you need to have answered are in your network, the real network, which is people, one-to-one. You can access Spela professors here. You have a unique chance. And it's not that you have to understand everything. It's rather about, you know, resolving some of the issues that perhaps you have and you or we don't get through the scientific language. Finally, is the patient language that matters. It will be very simple interactions with your kid that perhaps we will be able to solve. Not just perhaps, we have a really great chance. Risks are still here. Let us be aware of that. Thank you very much.
Thank you so much. So my name is Laurent Servais. I'm professor of pediatric neuromuscular disease at the University of Oxford in the UK. That's not where my typical British accent comes from. I'm basically from Belgium, where I still work. I'm super happy and excited to be here because I'm actually not a CTNNB1 doctor. I'm a muscle doctor, but I've been captured by Spela. She took me in the field to bring a little bit of my experience in building a clinical development project. So that's why I'm here. So apologize for understanding and knowing nothing about CTNNB1. My field is just about how to try to make it happen, right? So first, I got a couple of disclosure in the field of digital technology. Let me tell you why we use these little watches. It's because we want to be free of the tyranny of the single point. What do I mean? Let's imagine that these are two patients or two populations of patients, and obviously you have done something. They are diverging, right? But according to when you consider the endpoint, you could say, okay, there is no difference, but obviously the curves are diverging. So if rather than having a point assessment that you can do in the hospital or in another room, we could have the area under the curve, and we could have a broad representation of what is happening in the real life of your kid, then we could be much more powerful from a statistical perspective. I was a baby myologist several years ago, and I was working in Paris for my first day at the Institute of Myology. I was so impressed by all the science there. And I was looking at a patient in a clinical trial phase 3 running a 6-minute walking test. You know, this stupid test in which we ask the kids to walk in a corridor during 6 minutes. And I was so impressed. And then the mom told me, well, he's not doing well today. I was sad. And I said, well, maybe he's on placebo, maybe the drug doesn't work. What's happening? And she told me, yes, he's not doing very well because yesterday we went to Eiffel Tower and we visited Eiffel Tower. So we were gambling decades of research, million on investment on kids who were tired because they visited Eiffel Tower. Guys, you know the point. I mean, you've been traveling from Spain, from Poland, from Mexico, from the US, from Belgium to this hub, which is Slovenia. And I'm pretty sure your kids are tired, are exhausted. You are tired, exhausted, and things that we measure can be impacted by the fact that you are just not at home, right? And we're just making a point assessment. So how could we be rid of the Eiffel Tower syndrome? Well, first possibility to go to Brussels and to conduct clinical trials in Brussels, because there is no way to be exhausted in Brussels except for beers drinking and fries. That's where you can see how different are the countries in Europe, right? The French, they have a big tower. The Belgian, they have a small man peeing. Come on. So that's why in the entire field of medicine, we used we are using more and more wearable device. Rather than paying physiotherapists and doctors, giving a wearable technology to patients. And actually, it is more and more used in medicine. So that's what I'm doing for the last 14 years of my life, but certainly not alone, with a company that is called Sysnav. And after me, a lovely engineer from Sysnav is going to explain you the practicality of that. So we've been developing over the years a device. At the beginning, it was ugly, awful, and terrible. It was called an Actimile. And then we've worked on it, and it's now, you know, it's okay. It's what now we call a side, right? It's actually smaller than an Apple Watch. And that's the kind of signal that we can actually record. Look, this is a patient walking at home. The patient falls. The patient stands up. And the patient is able to walk again. We are able to record that at home continuously. It means that nowadays, we don't need to count the steps in the corridor. We don't need to examine the patients. We can just give a device and say, live your life. I don't care. Do whatever you want. I am able to measure every single of your strides, to identify your strides, and to measure it. So Melissa will explain you how it goes then in the cloud, and then we analyze it and we measure every single strides. So you could say, okay, fair enough, but this is very variable, right? According to when it rains and so far and so on. And this is true. When I was in Paris, I used to develop the theorem of Servet in Paris. When the French are on strike, they walk more, obviously because of public transport that does not work. Then I moved to the UK and developed these new theorems. When it pours, British people walk less. When it simply rains, they walk more, right? So yes, obviously, activity is social dependent, weather dependent. But let's imagine this is the activity of a kid. And parents are divorced. So during the week, he's with mom, and she's a sport lady. She runs and she makes sports. And then he goes to the cousins, the father's side, the mother's side, and they walk in the countryside. And then he goes to the father, and then the father is interested by barbecuing and TV watching. And then he goes to the cousins, the father's side, right? And then they just look at the Euro Cup of football. And I think that Belgium will win, by the way. Variability is huge. I agree. But if we average on a 3 days period, the variability decrease. If we average on a 7 or 9 days period, the variability decrease. So the longer is the period on which we average, the lower will be the variability. Well, it's not a joke. I mean, we could get approved by European Medical Agency, first as a secondary endpoint in Duchenne muscular dystrophy, then as a primary endpoint. And it is the very first time that a regulatory agency qualified a primary endpoint that is a digital outcome. That is something that we can do entirely remotely. So to accelerate things, we developed 2 protocols, and they are umbrella protocol, ActiMocap and ActiLiège. And 5 kids with CTNNB1 mutations came to Liège in the ActiMocap, because the first thing we want to do is to be sure that when we measure a stride in a patient, we measure a step, this is actually a real step, and that we are able to measure this step, right? So this was a control environment study, and we could confirm on these 5 patients that indeed we can capture every step of a patient with a CTNNB1 mutation, and we can indeed measure this step. So this was in Liège, Belgium. So now we are ready to move in the real life, and 20 of you will participate in the study, those with kids who are ambulant, so that we can now check in the real life outside the lab if we can measure it. And I'm going now to give the floor to Melissa, and I just want to finish by thanking my teams. This is my lovely team in Liège, and this is my PhD student. She makes all the work. I speak and she works. And this is the lovely team of CISNAV. Especially, I want to give the credit to Melanie, who was a fantastic, amazing person, dedicated to patients and to person with rare disease, and she died, unfortunately, last year. She developed a lot of work, but I know that her energy is with us today. So with that, I give the floor to Melissa. Thank you so much.
Hi, everyone. There is no slide. Just in the slide.
I think it's the same
Not working.
presentation.
This is the problem with
okay.
engineers.
Yeah.
There you are. And that's for pointer. So to move on.
Yeah.
To
Okay, perfect. Thanks. Hi, everyone. So I am Melissa Mena from CISNAV company. I am study manager. So I will be the main point of contact for this study. And I will just talk about the technical part of the device in the study. So as Laurent explained it, so the side device is just one station with 2 sensors, and both sensors will be worn on each ankle for the patient, for the ambulant patient. And in the suitcase, so there are the station, the devices, the sensors, the power cable, and the Ethernet cable. And the Ethernet cable will allow us to recover the data on a daily basis. So that means that we can check the data on daily way. So on the device, there are 3 LEDs for the just to know the charge part of the sensors, and to be sure that the data has been uploaded. So there are 3 LEDs on the station and 1 little LED on the side of the sensors to be sure that the device has enough charge and to be sure that the sensor is recording. So on a daily use, in the morning, so this is just a typical day of a child wearing the side device. So in the morning, the 3 LEDs on the station should be solid green, and the patient would just put the sensors on each ankles. And the little, as I say, LED on the side of the sensors, it's blinking in white. During the day, the patient will record as much as possible to record the data, and as Laurent explained it, during his activity at school, during sport, anything. And during the evening, when the patient will go to the bed, he will just put back the sensors on the station, and the little LED will start blinking green. And during the night, sensor will be charged and data will be uploaded. Regarding the positioning in this study, so patient will wear the sensors on each ankle, on the right and on the left, and there is little stickers to determine which one will be on the left and which one will be on the ankle. This is very important to keep the same positioning during the whole study, to not reverse the sensors, and the sensor will be just worn just above the lateral malleolus on each ankle. Another thing very important is the sensors are not waterproof, so they have to remove the sensors for any kind of water activity, and also for specific medical exam as MRI or CT scan. And just they remove it, they put it on a flat surface, because if the patient, if the parents put it in their pockets, that will record the activity of the parents. So just on a flat surface, immobiles, that would record inactivity. This is not an issue. And after, they can rewear the sensors and start again the recordings. Regarding the protocols, so they will start the recordings after this conference. So for 30 days, they will start to record. And after the 30 days, we will recover the data, give the family some feedbacks, and they will have another recording period here at month 12, 1 year after, of 30 days as well, just to know to see the evolution between the first recording period and the second one. So this is to see the evolution of the disease. There is only one thing, is that the sensors need to be charged every 6 months. So I would just send a little reminder to the parents to connect the station and to recharge the sensors. And for patients without internet connection, so there is a little USB drive in the station. So all data will be stored in the station, and we will recover the station just after the first recording period. And at the end of the study, we recover the device. I will be the main point of contact for this study. If the family has any questions, so there are the patient documentation in the suitcase. I will organize some training session today and tomorrow and we will equip the patient with the sensors. And this is my email address to contact me if there is any question. And you can also contact the investigating team if needed. Thank you so much for your attention, and I will be pleased to answer your question if you have any.
Thank you very much. But wait. If I'm reading the timetable correctly, then our next would be Thomas Carroll, whom we had already. And this leaves us with Dr. Laurent Servet again. And that's why I wanted you to stay on the floor. Melissa, thank you very much. So your presentation is called Managing Expectations with Gene Therapy. As said before, managing expectations is important. We have to know what we're looking forward to. So enlighten us.
Okay, thank you so much. So I'm the same guy, same accent, same origin, and many more disclosures in the field of gene therapy. I've got my fingers in many pies. Okay, I'm going to talk about things I know, right? It's easier. And I'm going to talk to you about two diseases that I know very well and that are very exemplative of what we can understand from gene therapy for our community. I say our community because now with Pila and Damien, I feel part of this community. Let's speak about Duchenne muscular dystrophy. And rather to show you data, I'm going to show you a patient. Can we try to get the two videos together? Any chance to get the two videos together, the technique? Do your best, guys. Yes, look at that. And now the other. So it's the same kid. On the right side, he has been treated, right? On the left side, it was before treatment. And look how faster he goes. He left behind because of the videos, but now he is going much faster in the stairs. It's easier for him. Is it a miracle of gene therapy? Not at all. It's a miracle of steroids. We just give him steroids, right? So this kid has not received gene therapy. He has received steroids. By the way, you know that when we give gene therapy, we also give steroids. So the reason why I made the provocation to start with these videos is that before dreaming about gene therapy, before dreaming about all this innovative medication, we need to have good standard of care. It doesn't make sense to start thinking about all these fancy approaches if we don't go for basic standard of care. So when it comes to Duchenne, there are plenty of things, but for CTNNB1, I mean, of course, we need to be sure that to take care of the contracture, to take care of the dystonia, to take care of the epilepsy, if any. So it's very important to maintain good standard of care if we want to make some benefit of the gene therapy. Then in Duchenne, we started to work on dogs. And that was in my previous life. We developed a gene therapy for dogs who could be treated by exon skipping and then by microdystrophin. And I must say these are dogs that are naturally sick, right? So we did not create sick dogs. We tried to treat dogs that are naturally sick. And rather than showing you a lot of data, well, this is actually the construct that we did with the AAV9, a full construct of a microdystrophin. And I'm going to show you what it shows in these dogs. Can I have the video, please? So it was a year with dogs with an I name, Izmir. So no, the video. No, the video. Yes, the video. Yes, you can. Fantastic. So as you see, this dog as a muscular dystrophy is weak, is not able to go above the bar and to get the food, even if he's committed, he does his best. That's our poor little Izmir. And then this is ici, which means here in France. And as you can see, this dog has the same disease, but he has been treated by gene therapy and is definitely much more tonic and able to climb. Okay, so you can make maybe a double-blind randomized placebo study and try to figure out who is Izmir and who is ici. Obviously, it's quite clear that one dog has been treated and the other one has not been treated, right? So of course, as family, as a parent, when you see that, you say, I want my son to get a similar drug, to be like Izmir or like ici rather than the other dog, right? And these videos has been shown hundreds of times. And as you might imagine, it has fueled a lot of expectations in the field. And there are 4 drugs that have been that went to the clinical trials from Geneton, Solid, Sarepta, and Pfizer. Well, yesterday, Pfizer announced that the phase 3 trial using this drug failed. Double-blind randomized placebo-controlled study with a drug very similar of the drug that was so helpful for dogs failed. Solid was on hold during a time because of safety issue, but one could move is Sarepta. And Sarepta made a construct with the different types of AAV, a different construct, and it was published in 4 kids were published in a very nice journal, which is called JAMA Neurology. Excellent paper that shows that indeed 4 kids who were treated get a much better muscle tissue. And they came with nice clinical data, and we can see that the improvement in this case was major. We talk about 9 points on a scale of 34. You don't need to be a neurologist to see that. You can ask to a blind medical student, he will see it. Very exciting. It works in the dogs. It works in humans, phase 1. Then they went to a phase 2 trial. And a phase 2 trial is already a placebo-controlled study. And unfortunately, we could not see a difference between the placebo group and the treated group. because both groups were on steroids, like the boy I've shown you at first. And then they went to a phase 3 trial. And unfortunately, again, we could not see the difference between the treated and the untreated patients on the primary endpoint. So it brings me to the lesson number 1 that I've learned in Ljubljana streets. A man is not a dog, and even less a mouse. So it means that as a community, we can see amazing data in mice. We can see amazing data in dogs, but we are humans, and there is a big gap between dogs and humans. Of course, before going to human, we need to be sure that it works on dogs, that it works on mice, but it's not enough. And you may have very exciting findings in dogs or mice that are never translated into humans for one or another reason. So let's keep our feet on the ground. It is very important to have animal models, to have animal results. But it's not because something is amazing in animals that it will be translated in humans. Let's keep it in mind, okay? Of course, there are drugs that work. And now let me tell you a success story. And the success story is the story of spinal muscular atrophy. It's a similar story with the AAV9, the one that failed for Pfizer, and the full gene of SMN1. The first data were published in the New England Journal of Medicine. Spinal muscular atrophy type 1 is a terrible condition. The kids look normal at birth. They start having symptoms in the first weeks of life, and then they die within a couple of months, and they do not present any motor acquisition. Kids treated with this medication start doing much better. And rather than showing you a lot of data, as usual, I will show you videos. And I will talk about 2 kids. One who is born in a country where we have good waffles and good chocolates and good beers and good fries and good offensive football player. And the other one is born in another country. Let's call them Jack and John. They have the same disease. They have SMA type 1, and they have been treated with the same drug in my center in Belgium. So this is Jack. This John, sorry. So John is the French guy. French guy was diagnosed at the age of 2 months because he was floppy, and he was treated at the age of 4 months. And he survived. And not only he survived, but he is progressing. He's now able to eat the good French food. He became a painter. Look, he paints. And he's a sitting painter. He's a painter with a scoliosis, and he is a painter with a deficit of the upper limb, but he's a great painter. And nowadays, John goes to school, and he's a happy kid. He should be dead today, but he's a happy kid, smiling, happy to go to school, clever. He will never be ambulant, that's for sure, because we came too late. But Jack is born in southern Belgium. Can I have the video, please? And Jack, the difference of Jack, it's not beer, it's not chocolates, it's not waffles, is that in Belgium we have newborn screening. And so he was identified at birth and could be treated at birth. And Jack became a neurologist, and he is now able to take the reflex of his patients. Can I have the other videos of Jack on the right side, please, and the center of the. So he's not entirely normal, right? But he's doing so well in comparison. So a key message is, of course, the earlier we treat, of course, the better it is. It's not a matter of waffles. It's not a matter of chocolate. Also, I would like to. So there are ethical issues related to one-shot injection, because gene therapy is a one-shot injection. The first one is that parents and sometimes believe that one-shot injections means that it's a cure. It's two different concepts. You can have a one-shot injection and a treatment, not a cure, but a treatment, something that improves the child, that increase the quality of life, that allow our little John or Jack, I forgot because I mixed the name, of course, our little French man to go to school, to be happy, to be alive, but not to be normal. It's a treatment, right? So it's not because it's a one shot that it's a cure. So we need to make the difference between both. And then it's very important to manage the expectations at the community level. And that's what I'm doing right now. In this fantastic paper published in this fantastic journal, which is the New England Journal of Medicine, there were two kids. And these two kids, look, they are there, they are doing incredibly well, but they were super young when they started the treatment. And may I have the video? And this is, we're going to see, this is one of these kids, of this super responder. He is doing amazingly well for a patient with SMA type 1. If I am the father of a patient with SMA type 1, I'll say, I want the drug of this kid. I want my kid to be like that. My kid is in the wheelchair. My kid is bedbound. My kid is a track. He has also SMA type 1. I want my kid to be like this. The problem is that this kid was so young at treatment. He was actually identified by screening, right? So it's unfair to make people believe that this is a treated SMA type 1. This was a patient identified by screening and treated at birth. It is a miracle. This kid is doing amazingly well. He should be dead today. It is a miracle, but it's not a SMA type 1 patient who has been treated. So my key message for you as a community was first, a dog is not a human is not a dog, but also one patient is not another patient. Jack and John are different. This kid has been identified by screening. It means that something you see on the social network may not translate to your son, right? So let's keep very realistic about everything we see, everything we read. And we have exactly the same issue for Duchenne. We got a fantastic type phase 1 trial that did not translate into a larger trial. So then we need also to be aware of safety. Gene therapy is not, trivial in terms of safety issue, and you may see sudden, unexpected, serious adverse reaction. This is a second lesson I've learned in the street of Ljubljana. Well, safety events are not the one that you generally think about. I don't think that this is a patient of Dr. Osred Kar, who has received Meijing Damien's herbs. But just joke aside to say that in the SMA clinical trials, we could see some events that can be quite concerning, like thrombomicroangiopathy. It's very rare, but we need to be aware of that, and we need to have a good safety plan because, and this is a third lesson of Ljubljana Street, it's good to hope to be lucky, but it's even better to have a proper risk management plan in place. So it's good to ask the dragon to give us the luck, but it's maybe not enough. We also need to think and to properly prepare our trials by getting the right tox safety and then to have a right design of our trials to be sure that collectively we are ready to manage any safety event that may happen during a trial. So then comes the problem of the risk perception. This is me with one of my patients. Let's imagine that I've got a drug that is very efficient, that could help him to walk, but that can cause 10% of problem. My patients could think, with this drug, I have 90% of chance to go better. And I would say, well, if I treat 100 of my patients, I could kill 10 of them. So the perception of risk is very different between doctors and patients. When you think about risk, you think about one patient, your son, your daughter. When we think about risk, we think about all of you, right? So sometimes we have a different way of understanding what means a risk. And so my two last slides, I've left Ljubljana to go back to Denver. Gene therapy presents foreseeable and unforeseeable challenges, but it can operate miracles. And let's remind the smile of this kid, and let's make the miracles happen all together. And all together we'll do it as a team. So here's my team in Oxford, but I feel really now part of a team, and that's together that we can operate miracles. Thank you so much for your attention.
Thank you very much. And I will ask you to stay on the floor again, and you can take a seat, one of those. So Damian, will you join us for a discussion? So we have roundtable discussion, I guess, with the people who were participating in the lectures that we had now. So I would also invite Melissa, and I would definitely invite Spela if she's here. And if I'm missing somebody, I don't know about the people in the video conference, if they're joining as well. But I would say the more the merrier. Have I forgotten somebody? No. Can I pick somebody to come up to be a sparring partner? Would somebody dare? Yes, the guy showed like this. You nodded, you're invited. Come. So what's your name? Ahmed, hi, from Canada. So you're welcome. One more. So who will nod now? Who dares to nod? Nobody dares to nod. I will invite Tanya. You, yes. There's a reason for that, please.
So Ahmed and Tanya, you take the difficult questions and we take the softballs, okay?
Yeah. All right. Thank you very much for this very, very clear presentation of science, Laurent. This is, and also a little bit of culture. And also for doing the plugs for Slovenia. I shall stop with that. I still have a few to tell, but still I would say that managing expectations and knowing about risks is basically a combination of two things. One is emotions, but there is also rationality. So we all are balancing. It's not that it's just the task of these people here, but it's actually our thought processes, our information, things we learn. And for all of us that work, let's say at the Institute of Chemistry, from my personal perspective, it's a learning, those are very steep learning curves. And science is very gratifying from that, in that domain, I would say. But since we have two new participants here, I will ask you to first introduce yourself. So I think Tanya is a scientist as well. So it's good that I start with you and give you 2 minutes to think about what you will tell about yourself, but be yourself. Is that okay? Tanya, you start. Tell us about yourself a little bit. Perhaps it's, yeah, I don't know.
No,
it works?
Okay. Yeah.
It works, it works. Thank you.
Okay, so I'm Tanya Zdolšek-Draxler. I'm also from Slovenia. Yes, I do work in research, but in the IT domain. But I also have a child with a rare disease, namely Kleefstra syndrome. I'm a patient advocate for Kleefstra syndrome. Yes, and that would be all.
Yeah, but you did organize a conference for the Kleefstra syndrome recently.
Yes,
it was a
last
little
year.
bit smaller than this one. So can you just tell us a little bit about that?
Yeah, so it was a little bit of a different setting because it was really like a purely scientific conference. So we really wanted to kick off the collaboration between researchers from all around the globe. And I think that we somehow succeed. But we are not as active as a community. So we are active, but more in a digital world. But our collaborators, families from the States, they do have yearly family conferences. So they are more oriented to this family gatherings, and we here in Europe more on the scientific side.
Thank you very much. Ahmed, now it's your turn.
Hi, everyone. Nice to see everybody. Our son Zain, he was born in 2015, so he's 9 years old. He was one of the earlier kids, I would say, that became part of this study. And so he's been part of this program since for a while now. And it's great to be able to see the progress that's been made. It's fascinating. I know the world that a lot of the research world moves very slowly, generally speaking, in the medical field, and it's fascinating to see how much progress that Spiela and everybody who's supported her has been able to make. We come from Canada, so we had to travel a long distance to be here, and we always struggle, in Canada, in the Ontario where we live. In terms of with CTNNB1, we always end up having to educate the doctors, believe it or not, about it more. So we know more about it, I think, than a lot of the doctors that we interact with. And I don't think that's probably something unique to where we live. But it's great to be, in Europe. I think there is definitely a lot of great work that's happening. And but we definitely struggle with that in North America, at least in Canada. I'm not sure about the US. Hopefully we'll see some traction there and maybe some conversations these 2 days in terms of how we can get some more collaboration done across the ocean.
Thank you very much. So Damian, I will. Damian, tell us about this issue about patients who need to educate doctors about a disease. That's not uncommon. That happens, right? Because you can't be an expert for 6,000 rare diseases.
Yeah, it's exactly the case. And I mean, I honestly can tell that when I first met Spela, I knew nothing about the CTNNB1 syndrome. So I learned on the go with her and, through everything that's been written since we started exploring this. And it's the same story with each other, any other rare disease. And, in my outpatient clinic now, it's not uncommon that a parent would come and say, we also have a rare disease. We also would like to have a story like Spela's, and it's just impossible to, to understand all the diseases to the extent that I understand now CTNNB1 syndrome. And of course, it's also not possible to, to engage in, so many different other diseases. We still try to help, so like SATB2, for example, is a disease that we have a mother here who is also a representative for the group of patients, and we try to go with the guidelines, but of course it's impossible to care for all that. So I think as doctors, we rely on our patients, and this has been a very fruitful partnership in many different cases, like in SMA, for example, and DMD. When the new treatments were available in the States, it took quite monumental efforts to bring this kind of treatments even before they were approved in Slovenia to our patients. And it was only by the help of patients that we were able to, to step over some major obstacles on that path. So I think it's very important for doctors to be open to collaboration, to say that, it's not uncommon and it's nothing to be ashamed of if you don't know a particular thing, but there's a lot one can learn. And by having a collaboration with patients, you can, you can bridge that.
All right. Thank you. So it's actually collaboration. I understood this interplay between the patients, between the patient advocates who get to know a lot, like Spela, like Tanya, like Erika, and others. So my question is to Melissa. Tell us a little bit about your work besides the sensors. So what is your interaction with patients, and how do you see the topic that we are addressing now? It's a learning curve. It's also, a lot of unknowns for patients. How do we get them on board? How do we create this communication channels? And how do we nourish that? Can you comment from your field of work?
Yeah, this is the first time actually that I am in contact directly with the patient, because normally we pass through the clinical sites. So we train the clinical sites to the use of the device, and after they will train the patient and family to the use of the device. So this is the first time for me. So this is not this is a good thing and very interesting. But normally the link is with the clinical site and not directly with the patient, unfortunately.
Yeah, but actually I'm not surprised, simply for the fact that a lot of things are to be learned, and some are in natural sciences, other ones are just in, how to communicate with patients. And this, I would say, in the work we do at the CTGCT, so Center for Cell and Gene Therapies, we found out that we are addressing three very distinct audiences. One of them is researchers, which is the native audience for the National Institute of Chemistry. So I would say this part of the whole, right? Then we have clinicians, which Damien, for example, you represent. Laurent, would you see yourself as a researcher or as a clinician?
I'm an hybrid.
Once more?
An hybrid, a mixed of two.
Good,
Yeah.
good.
Yeah.
So a hybrid, yeah. So
Hybrid.
So my Belgian English, which accent is that, is not really accustomed. So I apologize, right?
No problem.
All right, thank you. So, and the third group is, of course, patients. But we can't demand the patients to talk about AAVs and talk about third stage clinical trials and stuff like that. But some of patients have talent to do that as well. So you're not disqualified from participating, and also don't consider that you are forced to get to learn all of this, because a part of the story simply is about, connecting sometimes on an emotional level, sometimes, asking the beginners questions, but it's good to go along this path and get to know each other. So thank you also for going from the lab, from the clinical sites, going to patients with what you're doing now. So Laurent, you like dogs?
Yeah, I like dogs.
Yeah. You like mice as well?
No, I don't like mice.
Yeah, all right.
I'm not a cat person neither. I'm a dog person.
Right, all right. So what is the research that you're involved in, let's say, that drives you both happy and insane?
I think it's the very good way to ask me the question. So there are two reasons for me, from a professional perspective, to wake up in the morning and to be excited. The first one is actually this digital wearable device, because it has the potential for me to transform the way to conduct clinical trials and also to follow our patients. So I closely work with the CISNAF to develop a portfolio of digital outcome. And the second one is genomic newborn screening. So in my hometown of Liège, Belgium, we screen at birth for all treatable severe early onset condition. So we screen for 411 conditions at birth.
How many?
411.
Damien, how many do we do in Ljubljana?
So it's about 30, and now we started to screen.
30?
Yeah, 30. And but we now have started with genetic screening, and obviously the more good therapies that there will be out there, we will be able to implement more into the screening program. But
so we are following in the tracks.
Yeah, but Laurent is definitely leading the field with so many diseases.
Please continue.
So the vision is, there are many conditions that we could treat very early, but we don't screen them at birth because we don't have a good marker. So the vision of our team is we should be able to detect every single newborn at birth with a disease that we could treat as soon as possible. And the second aspect of this vision is to say, if we want to accelerate development in the rare disease like we did for Jack and John, we need to have a system in place that allows us, as soon as we have a drug that is safe, that has some target engagement, to be able to deliver it in newborns when it gets the maximum of efficacy. So our vision is not only to treat conditions that are treatable at birth, but also to be ready to conduct pre-symptomatic trials to accelerate and to leverage the efficacy of the drugs that have been demonstrated to be safe and to have some target engagement. We demonstrate the target engagement with our watches. So if we can modify, even if it's slightly, but that we can be 100% sure that we have a signal of efficacy, then it's enough. Let's move to young babies. Let's be as efficient as we can by moving to babies at birth. So that's our vision, to be able to treat the disease that are treatable today and to accelerate the development for the disease that will be treatable tomorrow.
Thank you. So there is one discrepancy that I can observe from the information that is being given is how many CTNNB1 patients there are recorded. There are about 400 around the globe, yet, Damian, you said that it's 1 in 50,000. If it's 1 in 50,000, this means that instead of this room, we would have to fill a stadium. So what's the discrepancy about?
I mean, you have to search for a disease in order to find it first. So with the novel genetic techniques, it's much more easy. Before, if you had a child who had a developmental problem and you could think of a syndrome that this child would fit in, then you would look for that particular disease. Now we have panels with over hundreds and thousands of diseases. And it's much more easier to detect a child with a particular disease. And also we did, for example, a small study in cerebral palsy patients in Slovenia. We looked at 150 patients with cerebral palsy who, and we excluded those who already had a genetic diagnosis and those who already had some tests done. So these were like really patients that nobody ever thought that they might have a genetic cause of their cerebral palsy. And we actually found 2 CTNNB patients in this cohort, so 2 brothers, actually twins. And so, I mean, we have to look for it. And I think if there's eventually an effective and safe treatment for the CTNNB1 syndrome, it will be included in, newborn screening programs around the world. And by that, I think the recognition of the disease is going to, lift off and many more patients will be discovered.
So we are underdiagnosed.
Definitely.
That's the realization right now. Ahmed, about your kid, I didn't record properly. Is it son or daughter? Son. So your son, what's his name?
His name is Zain.
Zain.
Yes.
Zain, when was he diagnosed? Don't worry.
Sorry.
Okay, don't worry.
So it took 2 years basically to get him fully diagnosed from birth. So we
2 years, 2 years, yeah.
2 years, yeah. He was born in 2015. It was a bit of a journey to try and understand, because as everybody else here, you start seeing some delays within the first few months, and then you start figuring out what's happening. So I guess we started his journey when he was about 3 to 6 months old. And we didn't actually get the full diagnosis until he was 2 years old.
Yes.
2 years. So, and they ended up having to do my wife is the medically inclined person in the family. So
right.
Sorry, I'm not
here with you, I guess.
She is right there, yes.
Raise your hand.
Yeah.
Thank you for coming.
So we did end up going for an extensive genetic testing that was very expensive, and it ended up being done in the US actually. And until then, nobody knew what it was called.
Yeah.
So when we got the testing back, we met with our doctor, and who was very a great scientist in his field, at least in where we live. And Canada has some amazing doctors as well. Yes. And he basically said, here's the diagnosis. There's all these papers that have been written about CTNNB1. It's a condition that consists of, this 1, 2, 3, no treatment. Good luck. That was
okay.
That was sort of
okay.
how we were handed off.
Yeah.
At 2-year mark. And I don't mean this in any negative way. This was the reality of what we were dealing with back in 2017.
Yes. I think it's the reality that we're dealing with very many rare diseases. So Tanya I didn't know how many people were coming to the panel now. but we are working together in a section for rare diseases. I think we also elected you as the president of the section. So what, in your view, is the problem of underdiagnosis? And also the problem of, here you are, I mean, it's your problem from now on. How do you deal with that? You know something about rare diseases at this point already, right? What's your view of the situation that we are in?
Yeah, so I think that we already mentioned before, there are so many rare diseases, it's impossible for the clinical side to know all of them. So it's just impossible. I don't know what to tell you. So I think that we are still lucky here in Slovenia to have a public health system. So this is something that I want to raise. In general, in Europe, we are quite good, but I'm always thinking also about the other world, so about non-Western countries. They have a totally different outcome there. So I would say that we still don't know enough about rare diseases, of course, and there are a lot of rare disease patients out there, but they just don't know that they have a rare disease. So what to say? So I'm still counting for the future to have a great benefit on is the digital health, so data. So I think that this is like beneficial, also sensors in clinical trials and similar. So and okay, you made all a great effort to come here physically, but this should be totally different. Families should be able to participate from homes, I would say. So we really need to change the mindset, the perceptives, and also the regulation, of course, because it all goes down to regulation. So what was the question?
I forgot.
Yeah.
No, you answered very nicely.
Yeah, there are really some, it's really such, there are so many questions. So it's really a broad topic to talk about. So it's not only one particular thing that we should focus on. So I really think that in rare diseases, it's particularly important. So data that are already mentioned, but also these relationships. Clinicians, doctors on one side, researchers, and especially important, patient advocates, patients. So to have this established relationship, because we really learn from each other, and if not working together, this will be a really slow journey. But we also see here in the CTNNB1 what can happen with when really dedicated people are stepping together. So, and they are building great teams. I can also say this for our Kleefstra syndrome community, which is in a way very similar to yours. Yeah, and all the others out there.
Thank you. Melissa, digital revolution in healthcare. Did it enter the clinical trials yet? How are things changing? You're using these devices to measure the gait. Are you measuring some other things as well with digital?
Not working. Yes, we can put the sensors on the wrist as well, so we can measure the upper limb movements. But yeah, the ambulance configuration for ambulance patients, it's ankle generally, and after for non-ambulant patient or for patient in transition, we can equip them with the sensor on the wrist and on the ankle as well, to have an idea of the movements that can be done and the power of the movements for the upper limb. But with the side device, actually, this is the only configuration that we have.
All right. Laurent, you use a lot of video in your presentations. Is this every conference or just this conference?
It's because I'm a very basic and simple person. I understand things when they are simple, and I understand much better a video than a graph with data. And for me, a video speaks much more, and a picture speaks much more to me than a set of data. I think that papers are the great moment to examine a set of data, but in a talk, when we have 10 minutes with people from different backgrounds, the only thing that speaks to all of us is a picture, is a video. So I try in general to avoid displaying data, and I think that there's much more to take home, to take back home in a video or in a picture. So short answer is yes, but it's just because
also in scientific conferences.
Yes,
you do.
Yeah, always. I've been told one day by someone that a good talk is a talk of which 50% of the audience will keep one ID 1 year later. It means that if 50% of the audience keep one ID 1 year later, it's a very good talk. If, of course, it's more than 50% of it, if it's 2 IDs, that's a lot. So we as scientists, we used to bring a lot of data, a lot of things to show that we work a lot. And actually, nobody reminds anything of whatever we display. So we need to be humble and say, what is my message? So I hope you got my message. My message is gene therapy is super exciting. We can do a lot of things. We just need to be cautious and try to understand what we're doing and not having crazy expectation. That was exactly the message I wanted to deliver. And that's why I've used all different types of videos and image and jokes and stupid jokes, just to show that, to say that. That was the only thing I wanted you to go back home with.
Okay, I was told once that people remember only mistakes and the emotions from a session. The message I got from you is that dogs are not humans. That was very clearly said, and I could go further, but here I would ask a few people randomly, and of course you will volunteer in such a way that I will show who actually is the volunteer. It's a joke. You still can volunteer. So what is the one message one of you got from this morning session? And mind you, it's 10 minutes before lunch, so good news is coming, in terms of food. So what is the one thing you got from today? Please. Yes, you can. We will come to that, and I don't know where the microphone for that is. We have a microphone is coming to you, and so let's wait. That's fine. No problem. It's fine.
Hello. I have a question for Dr. Damien and also Dr. Laurent.
Yeah.
As researchers, how many percentage do you need for a treatment to start clinical trials on humans? And would you eventually test it on yourself, with all respect?
So what is the risk ratio of
Yes.
a treatment? Is that the percentage you're looking
Yes,
for?
from when on you can say like, okay, it's 98% safe. Let's start clinical trials. It's 99%. So from when on you decide we're going to start clinical trials on humans.
Yes, yes.
And also, I really would like to know if you would test it on yourself.
Yes, yes, that is the question. Please, Laurent, you can start, and then
yeah, I can happily take the question. I live in Oxford, and as you know, in Oxford, they developed the Oxford vaccine, right? The AstraZeneca for COVID. And during the first day of pandemic, I received an email, do you want to be a volunteer for the vaccine study? And at that time, we had no data. It was the very first time that we infused this bloody vaccine. And I did it, not because I wanted to be immunized, because it was a double-blind randomized placebo-controlled study, but I did it for several reasons. The first one is that it was developed by my colleagues, and I wanted to show them that I trust them, and that the second thing is that I was sure that it was the only way to go out of this bloody nightmare of COVID, to develop a vaccine, that we could not do it without people to go in the trials. And that it was a collective effort, and that in a collective effort, we need to have serious investigators, we have to have serious plan, and we need to have serious patients. And as I was sure, 100% sure, that I was in the hands of serious investigators that had a serious plan and that were doing the things the right way, I think that we as a community were doing the right way by conducting these trials, and I step in as a patient. So the key part of my answer is I would do anything that makes sense from a community perspective, because a trial is a trial. It's not to treat a kid. It's not to treat me. I did not go in this vaccine trial because I wanted to get a vaccine as soon as possible. I went into this vaccine trial because I wanted a vaccine to come and to be able to meet again my friends, to meet again my families, to meet again people I like, and interacting with others in such events. And that's why I took part of the trial. A trial is not something we should do only for us. It's something that we should take part in because we act as a community the way we believe it's the right way to do as a community.
Thank you. Damian.
So I would start with saying that I believe in science. And as doctors, researchers now, we have, a slightly strange situation now where parents of certain, so parents of children who have a certain disease, they can see that, for example, FDA approved a medicine, it's not yet approved in Europe, and even without consulting with us, they would go to the States, and some of them, I mean, just recently, for example, would already get gene therapy, and actually a gene therapy that I personally don't believe have enough data to show that it's effective. So it's a very strange situation, but what I'm trying to say is that doctors are very cautious. I mean, as Laurent say, you don't want to harm your patients. This is the first kind of rule of any doctor. So you have to be really, really sure that when you want to treat a patient, you have the best kind of possible situation, and you can always expect some side effects, but you don't want to step on a stone that you already know is going to turn over. So I would like to say that we are doing our best here, to gain as much information, and us being here and doing the natural history study, and everything that came before that and everything that's coming after that. That's what we're trying to do to minimize the risk, to get the numbers as high as possible. But at the end, it's always going to be a leap of faith. It's always going to be, there's never going to be enough data to be 100% sure that this particular medicine is going to help your child, and there's going to be no serious side effects. So as I think Laurent really neatly showed another side of the story, but these two stories kind of come together. We try our best, we try to help the community. We will do our best to select the patients in the best possible way, because we know that some mutations are different than others, and we'll try to select those that have the least chance of harming. And and we'll do our best to provide maximum safety with the hope of some efficacy when we have the medicine. And if all this is kind of all the check marks are ticked, and I was in the situation of a patient or a parent of a child, I would I'm as Laurent, I'm a scientist, I believe in data, and I would go for it.
Yeah, but if I understand correctly, the drug, we have it today already. It's just not proven that it's safe. And it will take another year for us to convince ourselves whether the data tells the right story. Is that correct? Do we have the drug already as a candidate? Perhaps that's the question also for the researchers and some others. But the way I understand the pathways of when the trials started, we had mice trials, we had other types of investigation already. You're doing now this national what's the name?
Natural history study.
Natural history study. Eventually, it's not so much about whether we know where we're going, but we don't know whether there will be some obstacles, and we don't want to go blind into that. Is that correct? Is that the correct reading of that?
Yeah, I mean, when we started, you know, the whole story, I mean, it was like a very thick forest. We couldn't see through it.
Yes.
I mean, we didn't have any, you know, warranty that we're going to get through to the other side. Now, I think we're at the phase where we can see some light at the end of the tunnel. You know, there is some hope that a medicine that will be safe and effective is coming. So there's still some data that's missing, and that is something that we're gathering now in this 1 year that is coming, and maybe more, we don't know. But I think I'm pretty confident that we'll get there, and we'll have the answer. You know, this medicine is good or it isn't. But then if it will turn out that this medicine is not the one that we are looking for, I mean, we have other options. We have other, you know, stories that we're trying to develop. We just needed, I mean, as you have seen, you know, financial aspect of the whole story is quite important, because all this development is extremely expensive. So we could not, you know, start 3 stories at the same time. We had to decide for one, and we decided for a gene replacement therapy.
All right.
But we'll do our best. I mean, we will never go into something that we'll in advance know it's not helpful, or we would know that it's definitely going to be harmful for the patient.
Thank you very much. Thank you for the question. We have another question. Still, I will ask the lady who asked the question where she's from. What's her name, just to know? Where are you from? Thank you. Thank you for the question. I think those are the right questions we have to ask. Please, the microphone to the lady in the back. Introduce yourself shortly and then give the question.
Hello, everyone. My name is Tanya. I'm from the United Kingdom.
Yes.
I am a mother of an older child, so my daughter will be 16 next month. She is unable to walk unaided. She's in double incontinent, and she's nonverbal. She can say a few words, but mostly nonverbal. She attends a specialist school, but unfortunately, they're struggling quite a lot with her, to the point that she may not be able to go to school anymore. So my question is, for children, you know, for older children, what is, you know, hope for them really? Because from, you know, the conference today, from younger children, there is a chance that things can improve. But for older children, I just wanted to see, what is your professional opinion? How can life improve for them?
Okay, thank you. Who would take the question, Laurent?
I think that the very first thing is that we need to have a drug, a candidate drug, and one, to be sure it's safe, and two, to have some types of efficacy. It's foreseeable that the efficacy will be better in younger than older children, which makes perfect sense. I think that anyone saying that it will work equally, whatever is the age of the patient, is probably not understanding how a human brain works. The question is, what makes sense in terms of difference? And what are the expectations? For your daughter who is 16 years old with double incontinent, let's imagine a benefit that could be to acquire the possibility to go to the loo and not to be diaper dependent. It's not maybe a big thing, but it can be transformative, right? And I think that the magnitude of the benefit that we expect at different stage of our life is very different. If you speak to a mother today of a 1-year-old and you say the objective of the drug is to help the kids to be continent, you say, oh, this is not maybe super exciting. That's not what I'm expecting from a drug. So we need to make the difference between what we expect from a treatment and what we get from a treatment. And the magnitude of the benefit and the perception of what makes sense is very different if you are 1-year-old, 16 years old, or 15 years old. I'm not a parent of a kid who lives with a disability, but what my patients told me is that at an older age, sometimes a small benefit that can be the small problem that spoils life could make a big thing. So I would say it would be dishonest to say that a drug will give a normal intelligence to a patient with nonverbal who has behavioral issues at the age of 25 years. But it's still possible to get a minor benefit that may change life. That's what we've learned from spinal muscular atrophy. And interestingly, we could never find any correlation between the magnitude of the effect as observed by the doctors and the perception of the effect as perceived by the patients. Because we as doctors, we stupidly use scales, and even if we are more clever and more posh, we use magneto-inertial sensors. But the patients, they use their perception. And sometimes the perception of the benefit for all the patient, even if you cannot measure it, even if you don't perceive it as a doctor, can be massive. So my short answer is, actually, I'm pretty sure that the drug has the potential to give a benefit to the patient, maybe not with the same amplitude at different age, but sometimes with a similar impact on the quality of life.
Thank you. I think it makes sense to take one more question and then break. Is that okay with you? Would somebody like to ask the question? Some volunteers? There we have one. Thank you. The microphone is coming your way. Please introduce yourself shortly. Tanya, thank you very much for the question.
Hi, I'm Javiera. I'm from Chile.
Yes.
We're just 4 cases in Chile. My son Miguel has 7, and we have the diagnosis like 3 years ago. But it's very new. So I'm asked how important it is to look for more patients for the investigation to have I don't know, more financiation or better results.
Yeah, so if there are 1 in 50,000, somebody could do the quick check on 8 billion people. If that works out, I guess it's thousands and thousands of patients, if not hundreds of thousands. I don't dare do the calculation from my head right now. This is a question for Melissa, because you are dealing with investigative side. Laurent, you can take it. Yeah, please do.
I'm sorry, I don't want to take the floor, but I have a very clear answer. The more you are, the stronger you are as a community.
Yeah.
It's as clear as it is. Let's imagine that you have a drug that works. Do you really think that you will motivate a big pharma company for a drugs that affects 10 kids in the world? There is no profit to be made. If you are 2,000, if you are 5,000, if you are 10,000, you have a better chance. That is as easy as it is. And the understanding of the unmet need is the same at the national level. If a country feels that there is anyway no patient in this very country, why the hell should this country make any effort to reimburse a drug or to even think about reimbursing a drug? There are drugs that are approved in Chile in rare disease, in rare conditions like spinal muscular atrophy. So this depends on the number of patients you are and the interest of company at every single level. The possibility to get funding for research, the possibility to get funding for treatments approval, the possibility to get reimbursed depends on the number of patients. The more you are, the stronger you are. That's as easy as it is. If you want to make the community stronger, if you want to make it stronger as a group, be more numerous. That's as easy as it is.
All right, I will do the calculation now. 8 billion, I know you can't see it from the back rows, divided by 50,000 is 160,000 patients are in the world. Identified patients around 400. A long, long way to go. Thank you for giving a great question. Yes, we need to find more patients because the moment we find them, we are giving them hope in this direction. Hopefully we will achieve the goal, but there will be more supporters. And this is important. So thank you again for Chile. With how many people live in Chile? How many people live in Chile? So 20 patients per million times 7, you have 140 patients in Chile. Yeah, you diagnosed 4. So the discrepancy is huge. It might not be the right figure because this underreporting, I can't be sure about it, but very likely there is a lot of underdiagnosis, not just underreporting. And also, you know, it's hard to get the data in one place in this world, in spite of the fact that we are surrounded by Googles and Facebooks and all the other companies. It's very much about the cause. And I think the cause is clear here. So for the next hour, we have lunch. Please come back afterwards and we continue with the sessions. And dear panelists, everybody, thank you very much for participating. Thank you very much. Thank you, Melissa. Thank you. Thank you. Voilà. Voilà.
Recording stopped. One test. Okay.
Check, 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 check. 1, 2, 3. 1, 2, 3. 1, 2, 3. Yes, 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 yes. Oh, yeah. Take it back. One time. Check, check. 1, 2, 3. 1, 2, 1, 2. Bit of bass. Give me a bit of bass. Bit of bass. 1, 2. 1, 2, 1, 2, 1, 2. 1, 2. 1, 2. Yeah. 1, 2. Okay. Down, volume down a bit. 1, 2, 1, 2. Check. 1, 2, 3. Bit more. Down a bit. 1, 2, 1, 2. 1, 2. Bit more. 1, 2, 1, 2. Yeah. 1, 2. Yeah. 1, 2. 1, 2. One, two, 1, 2, 1, 2. 1, 2. 1, 2. 1, 2. Yeah. Okay. 1, 2, 3. Okay, mix me, man, mix me. Yeah. Start down. Whoa, whoa. Well, my friends, time has come. Raise the roof and have some fun. Throw away the work to be done. Let the music play on, play on, play on. Everybody sing, everybody dance. Lose yourself in wild romance. We're going to party, carava, fiesta, forever. Come on and sing along. We're going to party, carava, fiesta, forever. Come on and sing along. Come on, all night long. Yeah, next one, next one, next one. Next tune, please. Give me another one. Okay, give me another tune, please. He's deep into it. He's deep into it. Okay. Hola. Okay, give me another one, please. Molia man, do what I can. Won't you understand? Chapter 1 of Genesis, honey. Molia man, I'll do what I can. Won't you understand? Chapter 1 of Genesis, honey. I'll make no promises, darling. Got the bad heart and one lung. Y'all want me to whine and get down. So sex is a sweet and charming. Have mercy on me, my darling. I can hear my number calling. It's a blessing from above you're receiving. I'm only a man. I'll do what I can. Won't you understand? Chapter 1 of Genesis, honey. I'll make no promises, darling. Every jam that we jam, she wanna come stress me. Everywhere that we go, she wanna come test me. Dancing, vulgar, whining, your bumper making me heart just flutter. Teasing, squeezing, make me heart just raising up my blood temperature. I'm only a man. Okay, next one. Okay, next one, please. Wiggle wine. Give me the famous wiggle wine. Where are you? Okay, so the best. Okay, that one. Wiggle wine. Famous one. That's not. Well, it's the first two, and then see how the night goes. Play anything after that. Night long, wiggle wine. So go down. Or what? And then down. Okay. And just keep going on the list. You don't. Come on, everybody. Don't just take us on a case again, man. A little remix. You know it goes. All sexy lady. Pretty girl, wiggle and wine your body. Wiggle and wine, girl, you look sexy. Pretty girl, wiggle and wine your body. Wiggle and wine, girl, you look pretty. Pretty girl, wiggle and wine your body. Wiggle and wine, girl, you look sexy. Pretty girl, wiggle and wine your body. Wiggle and wine, girl, you look pretty. Switch, wind up your body, girl, raise it high. Wiggle up your hips, them, and take it to the sky. The music sweet like the food that you eat on. You look good, feel good, it's a state of mind. Show off your body, girl, you know you look fine. Sweet soul music will cleansing your soul. Your body is your temple, you never grow old. Wiggle and wine your body. Wiggle and wine, girl, you look sexy. Pretty girl, wiggle and wine your body. Wiggle and wine, girl, you look pretty. Go on. Wind up your body, girl, wine, wine, wine. Drop it low on time. Woo! Wind up your body, drop it low on time. Ah. One, two, sounds good.
Fuck. What's going on? What is going on?
Recording in progress.
Shall we continue? Perhaps another story, or rather not? Well, I don't know. I'm just asking. So I'm really enjoying my time here with you, and thank you also for reaching out. I hope you are somehow finding new connections. We are getting the right questions. I don't think that we should be afraid of any questions. You know, those are really serious things we have to address. We're talking about life. We're talking about decisions between life and, let's say, a life that is either better or worse, or even worse than that. We all know what that is about. I do believe in the power of the community. I do believe that I can't solve all the problems myself or prove to everybody that I'm capable of solving all. And neither do I believe this anybody in the room, but I do believe in this common direction. So if somebody's showing leadership at a certain point of time, it doesn't mean that that person will be a leader for all the future. So everybody can contribute. Sometimes the leadership is just a matter of a single day. There's an interesting story by Ernest Hemingway called, it's even a movie. It's called The Short Happy Life of Francis McCombray. It talks about a guy who was very shy, very afraid, and then went for a rhino safari. So they were trying to catch rhinos in sub-Saharan Africa. And there he caught his wife cheating on him in another tent. And then basically he found that he needed somehow to come clean because he was fearful all the time, and decided to be brave if somehow danger occurs. And there a rhino started stampeding against him, and he was the only one standing up, taking the rifle and trying to shoot the rhino. He wasn't successful. If you want to know what happened to the guy, talk to me later. Read the book or see the movie. There's a movie with Gregory Peck inside. Allegedly, my mother liked Gregory Peck, so she named me Gregor. That's exactly what she told me. I didn't make this up. Thank you. So we have a few more people coming now, and we will kick off this afternoon's part with a lady that I already got to meet earlier today. And her keynote is called Never Give Up, My Story of Success from Diagnosis. Welcome to, here in this Excel, I know you're Julia, right? Yeah, but it doesn't say on the same Excel that you're Julia, so I have to change the app. So to pronounce properly, you're Julia Taravella, an AGU mom, founder and president of the Rare Trade Hope Fund. Already the discussion we had in the morning was very insightful. Thank you very much.
Thank you.
The floor is yours.
Well, thank you so much. It's a great conference. I don't know about you, but I actually learned already a lot for our community. Just what I want to do is actually tell you a story about our disease. And one of the things, what I want to say, what all of you are doing here, lots of people asking me, you just a parent, you are just an advocate organization, which when I looked at that, what we achieved so far, and we're about actually to start clinical trial, probably. We are applying to FDA in the United States right now to start clinical trial. So what I call myself, and when I introduce myself to in different conferences or to different people, I called us in a nonprofit biotech, because all of you, and that's what we are doing, it's actually a nonprofit biotech. I did several master's degrees. I never was able to finish PhD just because I had to stop and actually take care of my children. But the acronym that I'm most proud of is RP. And I think all of you are here RP. It's a rare parent. my kids. I have two boys, Daniel and Alexander. It took me 18 years to actually get diagnosis for my kid's disease. When my oldest son, Daniel, was born, I remember the first sighting of the disease. I went to a play date with my girlfriends who had also boys exactly the same. They're all born within a week of each other. And it was 6 months. They were all about 6 months old. And both of the other boys already was crawling and tried to bubble something. And my child was barely able to sit up. And I was like, okay. And I remember start talking to myself, he's just a late bloomer, everything will come. And that was the theme of the story for the next 15 years. But then it was told to me by the doctors, he's just a late bloomer. So my poor child, Daniel, and then Alexander, but Daniel is much more affected because I noticed when Alexander was 6 months old, I really noticed exactly the same features. So Alexander is developed a lot more because I started all kinds of therapies on Alexander almost from like that 6 months old. I remember at 6 months, I will already put him in a swimming lessons. I already start doing stuff with him. I never did that with Daniel. And with Daniel, so Daniel has more severe disease. So that poor child, for the first 15 years of his life, underwent so much testing. He probably had 5,000 blood draws from all of the doctors. He had all kinds of MRI, CT scans, whole body x-rays, any kind of studies that are there, muscle biopsies, neurobiopsies, all kinds of stuff. Nobody can diagnose him. And finally, when Daniel was 15 years old, the doctors start telling me, okay, you have to make children happy and you have to stop searching, which really didn't stand well with me. What I did at that time, genetic medicine and actually gene sequencing was extremely expensive. It was the time when 23andMe was still $500 per person. So what we did, we actually budgeted our Christmas presents for everybody in a family and did everybody, we didn't buy any other presents that year. We bought 23andMe because that's what I thought, that maybe I can find something over there. But nothing came out, of course, from 23andMe. It was just the standard kind of ancestry and the standard results of that. In the next year, I took university classes and trying to find out and to learn more about genetics, because if it would be my children would be diagnosed, it would be completely up to me. So what I did is I actually download raw data, learn how to program, and wrote a program to identify what's the mutations my children had. And it took me about a year and a half. So I actually, when I run the program for the first time, and I remember it was February, I couldn't believe my results. The only gene that had kind of issues was the AGA gene. I read everything that I could, and probably like many of you, I actually felt into huge depression because I knew what is the result of the gene means, that there is no treatment, there is nothing that can be done for the child, and child is really is going to die within a couple of years of diagnosis. So the disease called aspartylglucosaminosurea. In short, it's AGU, but the foundation that we actually put up together, I couldn't call it aspartylglucosaminosurea cure. It would be really too tough for everybody to pronounce. And AGU is actually trademarked by American Geological Unit, which I couldn't do that too. So we called the foundation's Rare Trade Hope Fund. But AGU is the disease that has, It's a cellular protein. It's an enzyme that breaks down certain type of sugars. But as a result, it's autism-like disorder. So it is the language problems, is developmental delay and intellectual disabilities, skeletal abnormalities, and premature death. At that time, it was considered to be 25 years old. It is increased in the last 10 years that we're doing research to 35 to 45 due to ability to identify and actually manage the care of those children. We were lucky that I knew what was diagnosis kids had in February, but my husband completely was in denial first with my crazy idea. So he waited for the diagnosis in July from the doctors, official diagnosis. So our depression was, mine was finished by the time, and I already was in a full swing, and he just started. So we were very happy that our depressions was like 6 months apart. So we started gene therapy, and that was kind of, I screened for all of the possible ideas and people who can work on that. And gene therapy was the only option that's actually, at that time, was able to treat every single child with this particular disease. So what I will talk to you about is what happened during those times. So we started the gene therapy in 2013 and set up the foundation 6 months after I realized how much money it will take. The disease is extremely diagnosed because it is, I call it non-dramatic disorder. The children are very nice, and I am pretty sure that Laurent probably would like our mice because they're extremely nice and not mean. So, and that translates into children. Children are extremely nice. They're sweet, and you probably relate to that because most of your children are extremely nice, extremely lovable. And, but the children can walk, so it is less dramatic disorder. But as a result of that, we have a lot less diagnosed of the community. So it's extremely small community. From the time from 2013 to 2018, it was just me and one other family in France that was doing all the fundraising. So we started the research. We completed all of the trials that Spela did over here. And I think up to that, it took us exactly around the same time, around 5 years to get to that. And at that time, I was getting the advice from every single person, you need to find a pharmaceutical partner, which is a good idea because it's extremely, it takes lots of money and extremely important. And you can hear that all the time, get the pharmaceutical partner, find somebody will pay for a couple million for the development of the drug, several million for the clinical trial. And that's actually where our mistake was. We found that pharmaceutical partner. That's what happened in almost 3.5 years. So what that happened is the pharmaceutical partner came, they started the natural history study, they start production of the medicine, they looked at everything, they understand the gaps that's nonprofit foundation or nonprofit biotech can bring into the clinical trials versus pharmaceutical company, and they started to bridge those gaps. Remember what has happened in 2021 before the COVID, between the COVID and the economic downturn? You know what happens with ultra-rare diseases? That's a low-hanging fruit. That's the first things that gets dropped. Remember how around the world, every single ultra-rare diseases got dropped. And that's what happened to us. We were one of those people that's got dropped because it's non-economical for the bigger company to develop that. And as a result, we decided to pick it up and go forward. So in 2021, we started the drug production. So it is exactly what Spella is doing now. It is first you're producing the drug for toxicology, safety and toxicology study. And the next thing, you're producing the drug for your clinical trial. You can do it in parallel. And you're doing your GLP study. GLP is where good laboratory practices, it was where you prove that the drug that you produced is non-toxic in the animals. So you hopefully you translate it to people, to humans, and it will not be toxic to them. And that's where we're now. We're just about to submit all of the documents to FDA authority for us to start clinical trials. The protocol is developed. I think somebody was saying that there is lots of drugs that needs to be taken along with gene therapy just to support the gene therapy. Everything is developed. Most of the researchers are in place to go forward with the clinical trial. How much it costs us? It's about $500,000 to do everything preclinical and about $5 million to do everything from the drug production and toxicology study. It is extremely expensive. That doesn't include clinical trials. And that is where you need to community to come back. What we decided to do as a community, and it would be up to you to decide to do, we decided that until we pay for all of that, and we still have a little bit over $1 million to pay, we had $5 million. So we still have over $1 million to pay till this will be paid off. None of the children will be treated. So no children. So we decided to get together the community and do that. It's extremely important because you show that you're supporting your community, that you're doing the work, whatever hard it is. This is a table I want you to look at, and this is actually the key takeaways from all of the steps that we are done so far and we are doing. One of the things what I want you to look at is actually the cost range. And the cost range varies dramatically. It all depends on how do you do that, what innovative pathways you find in, and what support do you get. For example, one of the reasons, so when the drug is produced, let's take that as an example. When the drug is produced, there's lots of little steps that needs to be taken and lots of companies coming together. The main drug producer, from I understand from you, is ViralGen, but there's lots of little things for us we did it in lots of different companies. And one company said that we actually can do that part for you for free. If you will come with the children, and it was totally different country, you'll come with the children and actually show our employees what is it important for you and how our employees can support. You can reach out to the different companies within your country just to see whether they would be interested in supporting you. This is just one of the examples. And that's what's reducing the cost, or actually put that amount of money to reduce that overall dollar amount of that 5 million. It still will be 5 million, but there will be some additional things you can do to reduce that. And it's all comes from you all. Yeah, that's communications with regulatory agencies. Very, very important. I think Spella knows that very well. we have a great community, and you are actually even a larger community that is, our disease is actually a lot smaller than yours. But nevertheless, every community can come together and support. Gene therapy is something that can be treated every single child eventually. And so I'm just trying to, every single child can participate one way or another, be it a natural history study, on real treatment of the disease. Actually, what we're doing right now here is making sure that those parents that become parents of their child in 10 years will never have to gather here, will never to hear those stories, because they will be able to go to a doctor and get the treatment for their child once the child is born. Think about that. I don't know what will happen, whether my children will be able to participate in gene therapy, or whether only one child will be able to do that and another not. But nevertheless, I know that somebody will not go through that experience anymore. Well, thank you.
Lily, thank you very much. Thank you. We shall continue, but so the next speaker will tell us about the preclinical studies. I have an announcement to make. That's what I wanted to say in the meantime. And we will welcome Dr. Andrea Perez Iturralde. Is that correct? So the announcement is the gentleman in the green t-shirt, raise your hand. So that's the Canadian CTNNB1 Foundation. Anybody from Canada? Canada, well, that's a new name. So you should talk and try to get, people even from same countries. France, I know, is there. Other countries are there. Try to join in this conference. I know that, living hundreds of kilometers apart might mean that you didn't catch that people. Perhaps they are in this room. So I might do the same thing as I did with you. What's your name? Graham. So Graham came to me and said, please shout out anybody from Canada, meet Graham. Thanks. I'll do the same later for others. So the floor is yours.
Thank you. Well, the presentation.
It will come,
yeah.
I'm sure. Andrea, where are you from?
From Spain.
From Spain?
Yeah.
All right.
Yeah, but I'm working in Australia with LeChec, Children's Medical Research Institute.
All right.
We are working on the development of a gene therapy for CTNNB1 syndrome. So today I will try to introduce a little bit about gene therapy so everyone can be on the same page. And then I'll show you some of the results that we obtained in the lab. First of all, I'd like to introduce about genetic disorders. They impact more people than HIV and cancer combined. And this is quite surprising because normally people don't know much about genetic diseases. So actually rare diseases or rare genetic diseases, although they are rare alone, when they are combined, they are not rare anymore. Indeed, there are over 7,000 known genetic diseases and more than 8 million children are diagnosed worldwide each year. And many of them cannot reach the 5th birthday. So this is a huge problem. However, we are in an exciting time where we have been witnesses of a big revolution in genetics since the discovery of the DNA structure. Now we are able to modify the DNA. We can copy paste some parts of the DNA in other parts. We are even able to read the human genome. There was a project called the Human Genome Project that wanted to read the whole DNA in the human. of the humans, and it costed millions of dollars, and it took over a decade to do that, and now we can do it just in a couple of days, and it is way cheaper. So we have progressed a lot in these technologies, so we are in a very good time. However, although we know a lot about many of the genetic diseases, we have the tools to diagnose them, there is a big gap in which many of those genetic diseases, as you know, cannot be treated nowadays. And here's where gene therapy could help solve this gap. And what is gene therapy? Well, gene therapy, the simplest way to describe it is just the use of genes as medicine. So we basically, instead of using drugs, we use a piece of DNA that will be used as a therapeutic, and we can package them in what we call a vector, which will be like a delivery box that contains the address where the therapeutic should go in the body. And that's what we try to do with gene therapy. So instead of needing surgical tools, a surgeon, and new organs, many times with a vector and a therapeutic gene, we could solve or treat a disease. And this is, there are already some examples of this in the clinic. These three therapeutics are treating different genetic disorders for the eye, for the spinal muscular atrophy, and also for hemophilia B. And the three of them use the same vector, which is the adeno-associated viral vector, or AAV. And we use, we also are using this vector because it doesn't cause any disease in humans in the nature, and you can easily take out all the viral genes that it contains, and you can insert in there the therapeutic genes that you want to use. So this is another representation of the same thing. We can remove what's inside of the AAV virus and convert it turn it into a vector that will be able to insert the therapeutic gene in the target cells. So I'm gonna explain a little bit about DNA. Well, you all know, obviously, CTNNB1 is the gene for beta-catenin protein, and the DNA is like the set of instructions that we all have in the cells to be able to build all the proteins that we have in the body. So there is a special chapter that contains the information to build the beta-catenin protein. And this chapter is the CTNNB1 gene. And a single typo in this chapter could make the beta-catenin protein not to be produced properly. And that is what could lead to disease, because the functions of the beta-catenin protein will be dysregulated. And beta-catenin is involved in the communication between cells and also in the communication between cells and the muscles. And that's why many symptoms are associated with cognitive and locomotor defects. So with gene therapy, we can put, again, the chapter that has a typo corrected inside the box, inside the vector, and reach the cells that are affected, and then those cells will be able to produce again the beta-catenin protein properly. And we can also use some regulatory elements that here they are represented as tools that will allow a faster or more robust production of the protein. So when we designed the AAV-mediated gene therapy for CTNNB1 syndrome, we removed all the viral genes from the AAV vector, and we introduced the therapeutic gene, which is constituted of the set of instruction of the CTNNB1 gene without the mutation. and some regulatory elements that could increase the production of the beta-catenin protein where we want it to be, and also avoid the expression in other tissues where we don't want to overexpress beta-catenin. Two organs that we don't want beta-catenin to be overexpressed in are the liver and the dorsal root ganglion, and this is because it has been observed that the overexpression in these tissues could lead to toxicity. So we wanted to test if we could use these regulatory elements to avoid the expression of beta-catenin in these organs. So you can see the green is the production of the protein. So the more green you see is the more protein that is produced. And in the liver, we tested 2 different doses of vector, a low and a high dose. And in the dorsal root ganglion, which are the sensory neurons that are at both sides of the spinal cord, we used the highest one. And when we included these regulatory elements, we could avoid the expression of the beta-catenin. So we included these regulatory elements in all the vectors that we designed. So we designed a set of different therapeutic constructs that will include the healthy version of the gene and different regulatory elements, because not all the regulatory elements fit in an AAV vector. So we had to do different combinations to get the best one. And then we tested them in patient-derived neural models. These patient-derived neural models were generated from blood of a patient. These blood cells were reset. It means they were not blood cells anymore. They were pluripotent cells that could be turned into any cell type. We could turn them into muscle cells, into lung cells, but we wanted them to be neuronal cells. We wanted them to be neuronal cells because we want to know what's happening in the brain of the patients. And we wanted to use patient-derived cells because these cells will have the exact same mutation as the patient. So these neurons that we generate from these cells will act similarly as they would act in the patient. So from these pluripotent cells, we could generate 2-dimensional neurons, but also 3-dimensional brain organoids that are like mini brains that we can maintain in the lab, and we can test the therapeutics there. So we generated 2 different models, neuroprogenitor cells and brain organoids. We tested firstly the neuroprogenitor cells, and we wanted to check first that the neuroprogenitor cells were acting similarly to what we would expect from a patient, which is a downregulation in the beta-catenin production. So on the left-hand side, you can see a decrease in the expression of CTNNB1 in these patient-derived neuroprogenitor cells compared to the wild-type control cells that don't have the mutation. And we could also see, I don't know if you are able to see it, but we can also see under the microscope some differences between the wild type and the patient-derived neuroprogenitor cells. So a neuroprogenitor cell is supposed to have some projections because it is starting to differentiate into a neuron, and this is not observed in the patient-derived neuroprogenitor cells. So we are already starting to see some phenotypes that could be saying us that this is a good model for CTNNB1 syndrome. And then we also observed the dysregulation of a protein that is in close interaction with beta-catenin, which is the encadherin. So we checked that we verified that this model could be used to test the vectors. So again, we differentiated another set of cells, and we treated them with the vectors that contain the different constructs. And we pre-selected 3 of the AAV vectors with the 3 different constructs because they were able to lead to a higher increase of the beta-catenin protein, well, at the level of messenger RNA, actually. So these are AAV construct 2, 4, and 6. So we repeated another experiment with only these three constructs, and we observed this increase in the beta-catenin expression at the level of messenger RNA compared to both the untreated patient-derived neuron progenitor cells and the wild types. And we also checked for the function of beta-catenin. So we checked for the expression of DKK1 and MYC, which are two target genes of beta-catenin when it goes into the nucleus, and we could see a partial increase of the levels of these target genes. So we could see that there was also a bit of improvement in the beta-catenin function. After that, we move into brain organoids because it will constitute a better model because it contains not only neuroprogenitor cells, but also other cells that are present in the human brain. And again, we wanted to check that this will constitute a good model for CTNNB1 syndrome. So we also checked the expression of beta-catenin at the level of the messenger RNA. On the left-hand side, you can see again the drop in the expression in patient-derived brain organelles compared to the controls. And we also check it at the level of protein. So again, the more green you see is the more protein. And you can see that there is a significant drop in patient-derived brain organelles compared to wild type. And we also check for the expression of two other proteins that will indicate for the function of beta-catenin, synaptophysin, which is a marker for synaptic activity, so the communication between neurons, which was also downregulated in patient-derived brain organoids, and GFAP, which is an indicator, a marker for cells. And with this indicator, with this marker, we could check the activity of beta-catenin in proliferation of specific cells. And again, this was also downregulated in patient-derived organoids. So after that, we again treated the brain organoids with the different AAVs. And in this case, only the AAV that contained the construct number 4 was able to significantly increase the levels of beta-catenin messenger RNA compared to the untreated patient-derived brain organoids, and these levels were comparable to the wild type. And we also checked it at the level of protein expression. And again, you can see the difference between the wild type and the patient-derived brain organoids, and then how the recovery when you treat the brain organoids with the AAV containing the construct number 4. So with all this information, we selected the AAV with the construct number 4 to move forward into studies with animals, but Duško will talk about it later, so I'm not gonna talk about it now. But we also did a preliminary safety assay in a wild type mouse. This wild type mouse doesn't have the mutation in beta-catenin protein, in the beta-catenin gene. So we wanted to check if the overproduction of beta-catenin due to the inoculation of the vectors will lead to toxicity. So we inoculated the mice using the same route of administration that we want for the clinic, the ICV administration, and with 2 different doses, a high dose and a super high dose. The high dose is the highest dose that was used for the efficacy study that Duško will talk about. And super high dose is like double that dose. So we inoculated the animals and monitored them for 8 months. Weekly, we checked for the behavior, the physical wellness, and the weight. And we also collected the blood at different time points in order to analyze for toxicity markers or pro-inflammatory cytokines. But we are still working on that with the help of Duško. So for the moment, I only have these results, which is just the body weight, just for you to see that all the animals were growing similarly, which it is a good sign, because if they wouldn't, in that case, that would be a sign of toxicity. We did another sacrifice 1.5 months more or less after the inoculation to check if we could detect already the beta-catenin protein derived from the vector that we had inoculated. So we sacrificed a couple of mice, and as you can see on the right-hand side, we could detect already beta-catenin in the brain at the level of messenger RNA. This is 43 days post-inoculation, so we could already see that the beta-catenin is still there. So this is a good sign as well. We sacrificed the animals like 2 months ago or so. We took as many organs as we could, and we need still to analyze all of that. But when we were doing the necropsies, we also checked for any sign of tumor development, and at least macroscopically, we could not detect any significant observations of toxicity. So in summary, I hope everyone is understanding, but so we designed different set of therapeutic constructs that were packaged in an AAV vector and tested in patient-derived models, and we selected the most efficient. This vector was also tested for safety in a wild-type animal, and for the moment, it looks safe. And we also observed that there is expression of beta-catenin at least 1.5 months after the administration. And now we want to test this vector in more patient-derived brain organoids. We are also working on a further analysis for toxicity before going into the clinic. Thank you all for your attention. I'd like to thank, of course, the lab, because they all have helped me a lot during this journey, and also, of course, the CTNNB1 Foundation, because this wouldn't have been possible without them. The Little Miracles for Kids as well, the National Institute of Chemistry, and everyone. Thank you.
Andrea, thank you very much. You pretty much announced the next speaker. So about the same thing, but in mouse models from our Duško Lanščák. He comes from the, I would like to say, really phenomenal department for synthetic biology and immunology, and which I'm a part of with this CTGCT, which is a new project. Duško, happy to have you on the floor.
Thank you, Gregor, for your kind introduction. And by the way, you're doing an excellent job.
Thank you.
First of all, I would like to give a warm welcome to everyone, especially to parents who are basically fighting this battle. And I would also like to congratulate Spela for putting this beautiful event together, and also for being the driving force for this astounding thing. As already Andrea beautifully presented what they did, what a wonderful journey also they embarked on. I would like to present you basically our team effort in order to evaluate the effect of the leading candidate for treating basically CTNNB syndrome on a mouse model. You all know that in new drug development process, there are still crucial parts where you have to do some preliminary preclinical or non-clinical testing on in vivo in order to basically establish all the pipelines and also to get approval for human trials. And we usually use mouse model because of the great similarity of the genomes between the human and mouse species. And also they're relatively easy to work with, and basically also to some sort of degree, you can easily translate the findings from the mouse model to humans. In this story, we basically developed a new CTNNB mouse model. We then conducted a history study of animals where we basically characterized the animal, and then by connecting different research facilities, we also conducted efficacy study, and then also subsequently in vitro validated the therapeutic transgene effect of the candidate that was before presented by Andrea. We conducted a non-GLP, non-clinical or preclinical efficacy study, but we also have to take into account that animals will be also used for later on GLP toxicology study, which will then be beautifully presented by Jan afterwards. So getting and having a good animal model, in our case, mouse model, is quite important. So the colleagues at Jackson's laboratory basically developed a so-called heterozygous CTNNB1 gene model, mouse model, where basically they mated beta-catenin flocs with SOX2 mouse. That basically led to loss of function model, meaning that only one allele of the CTNNB1 gene is presented in these animals. And afterwards, when the genotype of the animals was confirmed, we then carried out so-called history study, or we characterized the animals with some testing. Firstly, we discovered that there's a significant difference in body weight between the wild-type littermates and CTNNB mouse model. You can see here differences for the males and females. So we are quite happy that we already observed some phenotypic changes in our mouse model. And then another simple test was carried out using so-called rotarot. Basically is a assay that basically determines the motor coordination or balance of the animals. You basically put mice on this rotating spindle, and you basically measure the time that allows the animal to basically stay on that. And we also discovered that at 12 weeks of age, we observed that for the males, that they drop more frequently or faster compared to the wild-type littermates, again confirming that there is a phenotype present in our disease mouse model. After obtaining this mouse model, we then went further and we contacted Charles River, basically a connecting point of the Jackson's laboratory, where they conducted a non-GLP efficacy study in order to basically pinpoint the therapeutic effect of the leading candidate that Australian people built. So they injected, AAV9 CTNNB1 vector into the mouse brain. Here, ICV injection, they injected 5 microliters of the virus, basically by doing stereotactic injections. And we use, as you can clearly see here, 5 different groups of animals, whereas wild-type animals were used as a control. And then the second group was basically heterozygous animal receiving placebo treatment. And then we use low dose, mid dose, and high dose of treatment for the animals with the appropriate CTNNB syndrome drug that is being now developed. The animals were injected at the age of 4 weeks, and then at the age of 12 weeks and 20 weeks, several different behavioristic assays were conducted, for instance, open field marble assay, kinematic gain analysis, and so on, to really pinpoint the therapeutic effect of the drug. I will just present you the main findings here because, as you saw before, many different tests were carried out. And I would just like to highlight the tests that showed statistical significance between the treated animals and non-treated animals. First test that was employed was so-called marble burying test. Basically, this is a very nice, cute assay where you basically have a cage filled with bedding material that should be familiar to the animals, that they don't afraid of this new bedding material. And then you just put marbles on that bedding material. And basically, this marble burying test determines the level of depression or anxiety when the animal encounters a foreign object, because these marbles are foreign objects to them. And you also basically can model compulsive behavior to those animals. And then after defined period of time, I'm thinking, we're talking here about 30 minutes, 1 hour, you basically just count the marbles that the animals were buried. And you can clearly see here that we observed statistical significance between the non-treated animals and treated animals, again confirming that we already observed a positive therapeutic effect on our mouse model after given the new drug developed for this CTNNB1 syndrome. The second assay that was employed, the so-called open field test. Basically, you put animal in an open cage, and then the camera basically films the animal's movement. And you can clearly see that if you have a so-called normal mice are very intrigued, and they basically investigate their surrounding. Whereas if you have an anxious mice, they don't like to investigate foreign surroundings, and they like to stick to the edges of these new cages. And basically this open field test is to use it to determine locomotor or anxiety-like behavior. And again, we were quite happy when we discovered that, there was again a statistical significant difference between the treated animals and non-treated animals, confirming that this drug is actually doing something positive. The last assay that also showed significant improvement, and we were quite happy about that, was so-called fine motor kinematic gait analysis, which is basically an automated high precision system to quantitatively measure changes in gait and balance, which are basically the clinical endpoints in movement disorders. And here you basically put animal in this corridor, like Laurent was before presenting for their device, and then the camera measures fine movement of the animals, movement of the paw trajectories, movement of the tail, fin limbs, and everything on. And then the computer basically translates those measures into definite scores. And here we were basically observing general gait pattern parameters, body posture and balance, and also fine motor skills. And we were quite happy when we discovered that there was again statistical significance at the age of 12 weeks, 20 weeks in all females and males for the treated animals, meaning that, again, positive effect in the phenotype, in the behavioral, in the motor function of the treated animals is present also here. And here we discovered that for the tail and back body posture, swing, and also paw trajectories, these are the crucial parameters that we observed that were improved. And we can confirm here that by doing 3 different statistically significantly important assays, we can conclude that this new drug has potential to be considered as a cure. And then afterward, at the end of the experiments, of course, the animals were humanely sacrificed, euthanized, and all the tissue were collected. And then qPCR was used to basically determine the level of the CTNNB gene expression from those viral particles that were injected in the mouse brain. And you can clearly see here a dose dependence in all brain tested regions, confirming that actually the vector is doing its job, meaning that the CTNNB gene, transgene, human transgene is being expressed in all treated animals. Okay, we observed that we could influence higher CTNNB1 gene production, which is basically the main thing of injecting the virus into the body of the patient or here animal organism. But we were keen on finding if that higher expression of the CTNNB1 gene can also result and translate to the higher beta-catenin production. If we observe higher protein production, because the proteins are those ones that are responsible for normal cell proliferation, normal cell function, which are basically the thing that drives you and also that are responsible for your life. And we were quite happy when we discovered that basically in all cases, we tested subcortex, cerebellum, hypothalamus, all different parts of brains. And we discovered that we observed higher beta-catenin production compared to untreated controls. And we were quite happy with those results. And we also confirmed, I don't know if you see this also by immunohistochemistry. Here's a picture of the mouse brain. And you can here see with yellow, the expression of beta-catenin protein. At the end, we also wanted to some sort do a so-called safety profile measurements. So we were wondering what happens with peripheral tissue. Therefore, we analyzed muscle tissue and liver tissue, as Andrea beautifully pointed out, that these are the off-target effects that you don't want to basically hit with your AAV9 treatment. And you can clearly see here that there was no effect of increasing beta-catenin production in this of this off-target tissue, meaning that this cure is considered here safe. And also we conducted a blood biochemistry panel. We observed, basically, we checked for kidney functions, for liver enzymes, for metabolites, and we didn't, again, observe any detrimental effect of the treatment of those animals, again, confirming safety or no side effects of the treated animals. So to summarize my talk, in this collaborative team effort, we basically developed a new mouse model to study CTNNB1 syndrome. We showed positive effect of the treatment. We observed not only higher DNA expression, we also observed what is also the most important thing, higher protein expression, no adverse effects were seen, and we can conclude to some degree that this is a promising clinical strategy for all the CTNNB syndrome patients. So at the end, I would like to acknowledge and thank all the people that are listed here. And of course, I would like to thank you for your attention.
Duško, thank you very much. We shall continue with preclinical studies. Ruth Buiters, where are you from? The Netherlands. Netherlands. Will introduce us to non-clinical safety testing for the CTNNB1 gene therapy. Please.
Good afternoon, everyone. Thank you for having me. I'm not on your schedule, so thank you for the organizing committee to squeeze me in at the last moment. My name is Ruud Buiters. I'm a toxicologist by training, and I'm currently working as a non-clinical consultant for a consultancy company called 3D Farm Exchange in the Netherlands. I got into contact with this organization only a few months ago, so I'm very new to your journey, but I'm very happy to be a part of your journey already and be here. I have a few minutes where I will try to highlight to you the importance of non-clinical safety testing, particularly the toxicology point of view for new gene therapies. And I think we already heard a lot this morning and this beginning this afternoon, but I'll try to give an additional perspective on that. So the things I want to cover in these brief minutes is actually, well, where does safety testing fit into the overall drug development, the importance of non-clinical safety studies. I also want to touch a little bit on the rigorous quality and compliance system that are involved. We already heard today mentioning of GLP toxicity studies. What does it actually mean, and why are we doing that? And very important also, what's actually the use of doing this non-clinical safety assessment? How do we use it as toxicologists? How do we work together with clinicians and regulators to try to get the best for our patients? So very broadly, I have a picture overview here of a classical drug development, which starts on the left in the light blue in a discovery stage, where you identify a target, in this case, where we look at the CTNNB1 gene, and where we try to find a compound that is efficacious against this. Like Andrea already showed, she tested a lot of compounds in order to find a treatment that can potentially be effective. On the other end, we also heard this morning a little bit about the clinical stage, which is more the red and orange part of this slide, where you indeed start your human clinical trials, treating patients with these three treatments. But there's like this little path in the middle, which is here in the darker blue, grayish area. It's not always a very popular part of drug discovery, because we are bridging the preclinical promise in the models where we show some efficacy to actually the patients. So we often are being seen as sort of a stand in a way, testing safety still non-clinically while you are desperately want to see some clinical results of these promising treatments. And I focus today a little bit on manufacturing and safety. I take them a little bit together because they are, for gene therapies, they are entwined. So what are we doing in non-clinical safety? We really want to ensure safety of the treatment prior to going into human trials. And what we want to do is we want to assess any potential risk so that we assure that the treatments we are giving to patients are safe and that the clinicians are very comfortable on dosing their patients with new treatments. In this case, and in every case already, it's not only about the therapy itself, you also directly characterize the whole production of the therapy. In this case, we are working with the biological. Andrea showed you have a DNA, you package that in a viral particle. There's a whole biological process involved in actually manufacturing that. And if you also try to do that at a little bit more larger scale, there can also be some unwanted process-related impurities in your final product. By doing those non-clinical safety trials, you also look at any effects of these type of process-related toxicity events. In the end, what we are doing, we are weighing all the data that is being generated non-clinically. And those can be laboratory studies, that can be in vitro experiments, that can be animal studies. And we build all that knowledge together to do a weight of evidence. And we make a toxicity assessment. We look at, okay, is this drug, does this cause toxicity? Also, where does it go in the body when we inject it? Does it stay in the brain? Does it move to other parts of the body? And whether we got any other responses to the toxicity. And I can already applaud my colleagues, Andrea and Duško, that presented that they already were taking a lot of consideration to safety measurements in the early efficacy studies, because that really helps already get confidence for a treatment before going to, say, pivotal regulatory required toxicity studies. So what are some of the safety risks specifically for this type of therapies? We already heard some of them today. And in the past, we have seen some toxicities with gene therapies, mainly being immune responses, toxicity in the liver, blood clotting toxicities, and sensory neuron toxicity as well. Fortunately, the field is moving very fast, and we get a lot of knowledge already also about what drives these toxicities. And sometimes it also means that we are able to avoid these toxicities. So as you may, have seen in Andrea's presentation, that she showed those green pictures of the liver and the dorsal root ganglia, where she showed very little expression of the beta-catenin expression. So that already shows some promise that we might be mitigating some of these liver and sensory neuron toxicities that are historically present in gene therapies. So compared to some other treatment types like small molecule drugs or some other type of biologicals, gene therapy, AAV gene therapy is considered relatively safe, but that does not mean that it is without risk. And what we are going to determine in toxicology studies is exactly what is that risk. And what we also notice that the regulators are also acknowledging that this field is growing and that the knowledge is increasing. And you also see continuous updating of the regulatory guidelines, giving you also the researchers a little bit more freedom to entwine toxicity and efficacy studies and to try to design these programs to be as fast to the patients as possible. I said I would touch a little bit on the quality systems and preclinical drug development. Well, there are two main systems. One is good manufacturing practice, which is abbreviated as GMP. And the other one is good laboratory practice, which is abbreviated as GLP. I start with the good manufacturing practice, which is a requirement for any clinical material. However, even though it's not a complete requirement for non-clinical safety studies, it is important that you already have your complete manufacturing process lined out. And what do we mean with good manufacturing practice? What happens in such an environment? Well, when a facility is generating a therapeutic product in a good manufacturing practice way, you can be assured that the facility was very clean, everything was hygienically handled, that production from different batches to different batches is very consistent, that all the staff is properly trained, that they keep records of everything that they are doing. So if something goes wrong, that they can search back in the records and find where it went wrong and find a solution. That they have all equipment that they use is very well maintained and very well calibrated, and that they also have quality control to see on all these processes that they actually happen. So it's not only there on paper, it's actually being controlled. Good laboratory practice is a sort of the same type of thinking, but then in a laboratory study space. So again, you have very organized and clean labs, detailed procedures, and properly trained staff. You have accurate record keeping, and again, also proper equipment and quality control. The importance of these systems actually that they give you a lot of assurance as a scientific field, but also the regulatory field who actually needs to give the improvements of moving forward from non-clinical development to a clinical stage, that the results you get from your data, from your studies that you're doing, that they are very valid, trustworthy, and that there's a better chance that they translate well and can be interpreted with confidence. So what do we do now with all these safety outcomes? Well, when we do these studies to identify any potential risk for adverse effects before testing in humans. And we have seen this morning that mice are not humans, dogs are not humans, they can behave differently. Not everything that we see in animals can translate to humans. But everything that we can already see and that we understand, that is what we want to inventorize. And the reason why we do that is that we want to give that comfort to our clinicians, to give them the confidence to dose their patients. And these findings, they will inform their risk management strategies in the clinic. So we can use our findings in non-clinical studies to optimize the dose towards the patients. We can optimize their monitoring protocols. So what are the things to watch out for in a clinical trial, to be alert on? And also, as also Laurent mentioned this morning, it's not only about if you can anticipate what kind of toxicities you might encounter in your clinical trial, you'll also be able to make a plan for it if it does happen. So if a toxicity would occur, then you are able to make a plan, and then you can act very quickly on treating the patient and trying to mitigate that adverse event. In the end, when everything comes together, it's always a risk-benefit consideration. So we try a non-clinical safety study, we try to get a best understanding of the risk as possible so that we can weigh the benefit together with clinicians, with regulatory agencies to find the optimal, to see if this treatment can continue into patients. So that's the ultimate goal, moving towards safe treatments into patients. So we try to do these non-clinical studies to protect our patients and our children and to ensure efficacy of the gene therapy. We apply these very rigorous principles like GMP and GOP to get optimal quality of both the material that we use, so the treatment itself, but also the study data we generate, so that we can interpret the data well and also translate that to the human situation. And in this case, I think the program aims to have both the effectiveness and the safety and use them hand in hand and try to get this treatment fastest way forward to the patients. And with that, I would like to thank you all for your attention.
Yes, we're listening a lot about safety, and safety comes first in medicine. The very known pledge that the doctors make when they start studying starts with the words, "First, do no harm." So we will continue with safety. I know it might be heavy on some of the audience, but it is important. So now, Dr. Jan Prochaska will join us. And I just followed you on Twitter.
Oh, cool.
Yeah. Well, I guess it was the CTGCT. So I guess it was you, because you were liking a post by the director of the Chemistry Institute, Gregor Anderluch. And very active on Twitter is also Dr. Roman Jarala, is the head of the department that creates all this magic here in Ljubljana at the National Institute of Chemistry. We're very happy to have you here.
Thank you very much. Thank you very much for such a kind introduction. And also I would like to thank Spela and organizer for inviting me and my colleagues to wonderful city of Ljubljana. Thank you very much for that. And now I will try to actually describe our part in this fascinating project. So I'm coming from Czech Center for Phenogenomics, which is large research infrastructure in almost Prague, let's say few meters from the Prague border, from some funding reasons. And what we are actually doing, we are as a research infrastructure focused on the work with the mouse models from actually level of the genome editing to characterization of mouse phenotypes and also to preclinical development. As I said, we are center just next to Prague, and you can see that we are not so small team. We have 150 people roughly, and actually 10,000 square meters place where we do research and keep mice. And what to imagine when we are talking about research infrastructure. So we are not company, but we are also not the academic lab. So you can imagine it like these wheels, and that we actually have our own research program, but we also provide the surveys, assistance, and everything is driven by capacity and the philosophy of open access, which is actually very smart philosophy, which actually was seeded by European Union to research infrastructures which were built throughout Europe, that basically anybody can ask for our capacity, and we are supposed to help or handle the project if our capacity allows. And this is what actually happens with CTNNB1 project. And that's why I'm also here. So we are not the local infrastructure. We are part of the infra frontier. So you might know the infra frontier is the European level infrastructure connecting all, let's say, mouse centers over the Europe. We are also part of the global consortium IMPC, which is working for full annotation of mouse genome. And also we are part of EuroPDX consortium, which is focused on humanized cancer models for preclinical research. To achieve that, actually my team is called phenotyping module, and we are distributed in a couple of units which are focused on different parts of physiological investigation. So when we have a mouse model, we are able to handle it from the clinical biochemistry to behavior analysis and everything in the middle. We have also a team for working with the infection models and the cancer models. And what's actually our let's say, direction where we are going is to move from the mouse genomics to understanding of human genes. So we are using our transgenic technologies to to also design and prepare we call it avatar models, where we introduce the exact genetic mutation from patients into mouse model, and then we can characterize and also design therapy or eventually use it for preclinical development. So, and to achieve that, we have highly specialized preclinical pipelines where we are trying to use maximum modalities which we develop on a background of comprehensive phenotyping for mouse genome annotation, and we are using that for testing of new drugs, but also drug repurposing projects. And last 2 or 3 years, we are also developing the gene therapy programs for that. And this is our local activity in the Czech Republic. So we actually are working in this consortium of, I would say, the most significant institutes in Czech Republic in the development of gene therapy technologies. So for my team is focused mostly on neurodegenerative diseases as Angelman syndrome, Prader-Willi, and skin-related diseases. But other partners are also following different types of rare diseases and we are trying to share our technologies and approaches how to. So, but now let's go to really hard reality, and that's what we need for this project, and that's to run the GLP based study for toxicity. And it was just introduced before I came here what the GLP is, so I will go quickly through. We can actually conclude here that it's just the standard which unifies the laboratory approaches and makes them comparable and reproducible actually between countries, between partners. Where does it apply? So you can see it's not just only for the therapy, but also for the cosmetics, veterinary, food, feed, and also industrial chemicals. So the GLP is basically almost everywhere where you need to get the product to a human or in a chain which is leading to human consumption. So why is that? And this is very important because you can imagine just if you heard the example that it's not pretty difficult to do a tox study in a lab. You don't need much, and basically it's quite straightforward procedure. But the GLP is here that the quality of tests is as high as possible, that the data are reliable and very important, specifically from the medical development, is the reproducibility of data and also the comparability of data. This is extremely important when you imagine to develop such a global product, like, for example, gene therapies. But what it means really from the perspective of somebody who has team who is supposed to work in a GLP. So you can imagine from this cartoon says basically everything. You can imagine that every part which is included in GLP, which means the facility, organization, all personnel, all devices, all reagencies, all material, all protocols, everything has its own paperwork. So, for example, recently we already reached the number of 180 SOPs, which are signed by authority and are regularly audited. And with this study and other studies coming, we will break 200 very soon. And this is something what you keep paperworking and paperworking. And something what could be done with one person, you realize that you need at least 5. And and this is basically never-ending story. But what is important, there is a need for that, and there is a good reason to have it. And that's why we are doing that. But what is extremely important, you can imagine the tons of papers which are behind. But what is very important is that every study must have very detailed study plan. This is critical. This is not just like waking up and saying, okay, today I will test whether it's toxic or not. You have to prepare the study plan in advance. And this study plan must be actually authorized by the, let's say, our National Drug Control Authority. and they can put some additional readouts or tests which they want to have in a tox study. So this is something also what we will need to clarify for beta-catenin study, but basically I think we are in a really good way to achieve this. So we need the consultation with the authority, and the authority confirms that the study plan is according the best of their practice. And now we are getting to a study which will be housed in our center. So here are the details of the studies. So we will have 2 cohorts. One will be short, one will be a bit longer. And maybe I can skip the boring text and go to cartoon. So so we will use GLP validated ICV injection in a stereotactic frame, where we basically are using the state-of-art technology to deliver the small volumes into brain ventricles. And then actually we will wait. We will wait animals. We will check their vision, and in a terminal phase, we will collect the blood samples, urine samples, and we will do detailed necropsy. So specifically brain, but all organs will be taken by 2 trained and audited pathologists, which will provide the detailed histopathological analysis of all organs which are actually in a mouse, actually. And the important is that there will be actually pathological evaluation of all possible changes. We should be prepared that there might be some background lesions also in a wild type animals, because we are working with the live natural system where is also certain level of randomness. So it can happen that, for example, wild control mouse can have a lesion in a gut, or for example, stone in kidney or something like that. And all of this must be reported and evaluated whether it's relevant to treatment or not. So this is probably the most tricky part, to have the proper histopathological analysis, but otherwise you can see that it's pretty straightforward design. The little tricky is the injection into brain, and the rest is very simple experiment. But just imagine how many papers must be signed to actually finalize this study. But we will make it. And here I would thank you for your attention, and I'm really eager to take any question to this problematic, if you have any.
You will get questions in the roundtable.
Okay.
Yes.
So I'm leaving.
Yes.
Thank you.
We will now listen to Mrs. Christina Martin, and she will tell us about manufacturing of hope. What are the projected timelines? I think she comes from Viral Gene. Is that correct?
Yes. That's
Thank you
correct.
very much. You're welcome.
Thank you.
Thank you.
Okay, so well, first of all, I would like to thank you for inviting us to this conference. It's amazing the work that you have done and all the work behind all the foundations that are here today. So I'm going to talk about the manufacturing of a hope, the manufacturing of CTNNB1 product, and the projected timelines. But I would like to start introducing myself. As he has mentioned, Gregor has mentioned, my name is Christina. I was working for Biogen for more than 3 years and a half, and I'm biotechnologist by training, and I am the project manager in charge of CTNNB1 program in Biogen. Okay, so first of all, I would like to start with a question to make this more interactive. So please, can anybody from the audience let me know what can you see in the screen? Zeros, yes, we have a bunch of zeros here. In fact, we have 13 zeros. So what happens if we change the first zero by a 1?
we are obtaining a very huge number, which is 1,000 billions. And for you to have an idea of the dimension of this number, we are talking about more than 1,000 times the people that are currently living on Earth, which is a lot. And probably you are wondering why I'm talking about maths and numbers when this is related to gene therapy. So it has a sense. This is the number that we need to produce by average for a patient to be treated with gene therapy. So this is the number of small vehicles. You have heard a lot about them during the previous talks. So these small vehicles, therapeutic vehicles, are the AAVs and contain the right piece of DNA, in this case, CTNNB1 gene, okay? So they are really tiny molecules, and we need to deliver this huge amount of molecules through the tissues to the cells that we need to bring the gene in. And in this case, these cells are part of the brain, okay? As, for example, Andrea has explained in her talk. So I would like to give you a brief introduction about our manufacturing process. So continuing with the parallelism of the vehicle, I would like to explain you our manufacturing process as a car, okay? So for that, we need the engineer. The engineer is the producer cell line, and we need to give the engineer the proper information to produce the AAVs, okay? So for that, we are using 3 different pieces of DNA that contain the information that we need to provide the cells to manufacture these AAVs. So we will use 3 different pieces of DNA. The first one is the packaging plasmid that contains information about the shell of the AAV, the outer part. So in this case, it will correspond, for example, to the number of doors that the car has, the windows, or the color of the car, okay? Then we have the transgene plasmid, that is the CTNNB1 expression cassette, that is the passenger that goes inside the car. So it contains the corrected gene and is delivered to the cells that we want to modify. And then we have the helper plasmid that is providing the tools that the engineers will need to build this car. So for example, the hammer, the screwdriver, etc. So we mix all these pieces of DNA, these pieces of information to generate a cocktail of DNA, and we introduce that inside the cells. So we call this process transfection, and we need to transfect more than 1 million cells per milliliter, which is a very high cell density. So we need some weeks to reach this cell density. So after the weekend, the cells are manufacturing our car, our vehicle, our AAV, and then we purify it and vial as in the format as you can see in the image, okay? So where we are doing all this process, so for that at Viral, we have 13 different GMP suites. I was going to explain what GMP means, but they have talked a lot about this term. So you can see in the image some of the bioreactors we are using on our installations. And these 13 GMP suites are located in 2 different buildings. The building you can see in the right side of the screen is the clinical facility. So here we will produce the different vectors that will provide to CTNNB1. And then if jumping into commercial, we have also the commercial facility in which we will manufacture the CTNNB1 product if moving to commercial, okay? So just to give you a bit of context about our company, it is a joint venture between AskBio and Columbus Venture Partners. So AskBio was providing the technology and the deep knowledge about AAVs and also about drug development, and Columbus was providing the funding. So it was raised in 2017, and in 2020, it was acquired by Bayer, giving the final boost to this project. So we are located in the north of Spain in a very beautiful city called San Sebastian. And now we are more than 400 employees. But as you can see in the next slide, we started just with 7 people. We call them the 7 Magnificent because they have done a very huge work. And over these years, we have achieved different milestones, but I would like to stress out the GMP clinical certification for the clinical facility and also the commercial certification. And in 2021, we opened the commercial facility that I was showing you in the previous slide. So through all these years, and thanks to the huge effort of our team, we were able to manufacture more than 1,000 batches. Out of them, more than 100 are GMP batches, and some of them are enrolled in different clinical trials, specifically in 27. We are working with more than 65 clients, and we are not working just with companies. We are also working with foundations, because for us, the corporate social responsibility is really important. It's a key part of our DNA, and one of our main objectives as a company is to work with foundations to help broadening the access to patients to these gene therapeutic treatments. So we are working with different foundations, as you can see in the image. Probably you have heard about some of them. We are working with Columbus Children Foundation, with Curious PG50, Phoenix Nest, Elpida, Hannah So Foundation, among others, and now with CTNNB1. So going back to CTNNB1 project, what we are going to perform, well, we have already performed the first step. I will show you later the results we have obtained. But we have manufactured, as I was mentioning, a 2-liter scale. When I'm talking about scales and liters, I'm talking about the cell culture with which we are beginning with. And in this case, this feasibility scale has a double purpose. The first one is to ensure that the product is fit into our platform, because here we are not talking about a predefined recipe or predefined chemicals. We are talking about complex biological systems. So we need to ensure that the product of its client is fit into our platform, and we are able to provide a good productivity and a good quality product, okay? And then the other purpose of this scale is to provide enough material for research studies. Then we have the 50-liter scale that will be intended for toxicological studies, and the last scale, that is the GMP scale, will be intended for the clinical study. So going back to the manufacturing process, I would like to zoom in a bit on the 2-liter scale. So probably you remember this picture that I shown at the beginning of the presentation. As I mentioned, this is called transfection, and it's triple plasmid because we are using 3 different plasmids, 3 different pieces of DNA. So then we are letting the cells manufacture these AAVs over the weekend. And then we continue with the purification of these AAVs. Through the process that you can see in the screen, we have several steps to obtain the final product, because the aim of this purification is to remove all the impurities that we can have in the product. Then this final product is stored below -60 degrees. So here are the timelines for this manufacturing. So first of all, I would like to show you the timelines for the 2-liter manufacturing. So as I mentioned previously, this, sorry, the letters are moved, but as I showed you previously, we have already manufactured this 2-liter research rate run. We were also working on the transient plasmid amplification in an outsourced facility, and we were able to already manufacture the AAV. Now we are in the quality control step, in which we are doing all the assays that we need to perform to ensure the quality and having a good productivity for this product. So we will end them on next week, and we will be able to close all the documentation that will be provided to the CTNNB1 Foundation. So here you can see as well the timelines projected for the 50-liter run and the GMP run. So these are tentative timelines, and just to let you know, as a rough estimation, we will start with the preclinical manufacturing, the 50-liter run by the middle of Q3, ending this manufacturing run before the beginning of next year, and we will start in the beginning of next year with the GMP run. So as you can see, we will perform the manufacturing of the 50-liter and then the QC characterization and revision. And as well for the GMP round, we will perform the QC characterization and revision. And this is taking a lot of time because we need to ensure the quality of the product and also to do all the documentation that my colleagues were talking about, which takes a lot of time. Okay. As you can see as well, we are outsourcing the transgene plasmid amplification for both scales. So I wanted to show you some of the results we have obtained for this 2-liter run. So on the right side on the screen, you can see the results for ITR-ddPCR. So this is a measurement that allows us to know the concentration of vector genomes that we have, and is a measurement of the quantity of AAVs that we are having in our final product. So here you can see the average of biology and historical data for AAV9s. And you can see that CTNNB1 product is performing well, since this is above the average of historical data that we have obtained. And also for the 2-liter scale run in the lower part, this is the ITR-ddPCR titer as well. CTNNB1 product is in the 70th percentile. So the same for the percentage of full capsids. This is the capsid that contains the complete expression cassette. So you can see that CTNNB1 product that is rounded in red is about our biology and historical data, and it is in the 62nd percentile. So I cannot end up this presentation without saying thank you on behalf of the whole Biologian team. As I mentioned previously, this is very important with us. We are very happy to work with foundations like yours. Here you cannot see the complete team because this picture was taken 2 years ago. But for us, it's a great source of motivation to work with foundations like yours and give sense to the work that we are doing in a daily basis. So thank you all for your attention.
So we see what a difference a single one does if you put it on the right place. So without the one, everything is a 0. Adding a 1 creates some kind of a result. Of course, we're talking about science here. Hopefully, when we do the roundtable, which is once after the next speaker, we will be able to touch on the, let's call it the human side. So prepare. All right? So the last but not least, is the person who is the referee. One of the referees, of course. She's not alone. She comes from the broad universe called the European Medicines Agency. Zuzana Vidic comes from the Slovenian Medicines Agency. The acronym is JAZMP. And we were very happy to have her on the panel we had at the end of May. So we realized that everything is progressing. Regulation is progressing. Experts have to learn. Patients are learning. So thank you for being with us again, Susanna.
Thank you. Thank you to organizers for inviting me. It's a big pleasure that I can give you some insight in the regulation of ATMP at this, I think, very important event. So just little to introduce myself. So I'm assessor at the agency, Slovenian Agency for the Medicinal Products and Medical Devices, assessor for biologicals, including ATMPs. So the organizer asked me to give you some insight also in the possibilities for the earlier access to these important medicines. But I may also give you as an assessor some perspective in this way. So if I just start from the beginning, the regulation on advanced medicinal products was adopted in 2007 already, so, and was assigned from 2008. So from then, these all ATMPs which are developed or should be marketed should get actually marketing authorization. The main aim of this regulation was to protect the human health and rights, and also on the other side, to allow that all medicines that are available, that these are available to patients across the whole Europe. So for this reason, the idea of regulation is that the marketing authorization of ATMPs can be obtained via a centralized procedure, which means the application needs to be submitted to European Medicine Agency. And this means it's a one application, one assessment, and for the successful applications, one marketing authorization, which is valid in all member states. So how does it work, this procedure? So for the assessment of the product, there are several experts involved, not only from the European Medicine Agency, but also from the national competent authorities. Those are teams of experts, which are team of rapporteur and a team of co-rapporteur who do the assessment independently, and then they get it together. During the assessment procedures are also involved experts as member of Committee of Advanced Therapy, and also from the Committee of Pharmacovigilance Risk Assessment Committee, and from the Committee for Medicinal Products for Human Use. So all these committees and experts from national competent authorities during the assessment procedure collaborate. And in the end, the CHMP is the committee who gives a positive or negative opinion on the medicine, and then this is sent to the European Commission, which makes the final decision. But marketing authorization is not an obligation for marketing of the medicinal product in the member state. This is the decision of marketing authorization holder in the end. So although there might be approved medicinal product, approved ATMP, but not necessarily to be available in your country. But yeah, we heard a little already about this. If we look from the product development perspective, yes, it takes very long time. It includes pharmaceutical and non-clinical development, phase 1 clinic and 2 clinical development, phase 3 clinical development, and then a submission of application. I have to say that actually very much effort, not only because we heard a lot about the non-clinical development, but a lot of effort needs to be put also on the pharmaceutical development, but development besides clinical development, because pharmaceutical development is very important from the perspective that the developers need to show in the end that the production process is consistent, despite the fact that for the ATMPs, we know that the starting materials or many raw materials are of biological origin, which means they introduce a lot of variability in this production process. But in the end, there should be kind of consistent process, and the manufacturer should end up with the specifications which are scientifically sound, and this should be for the identity, content, for the purity, impurities, for the biological activities. All these are important things. And I have to admit, as an assessor, that usually we end up, no matter whether it's a clinical trial application or is a marketing authorization application, most of the questions we end up on the quality part besides clinical part. On clinical part, there are many questions mainly related to the clinical study, selection of the clinical study population in relation to the selected therapeutic indication. Also on the selection of dosage, frequency of the dosing, on the treatment duration, all these kind of things, also on the administration route of application of the drug. So all these things needs to have some scientific rationale, which is of course also based on some non-clinical studies. So it is very important that drug development goes through all these phases if we want to have a product that is safe and effective in the end. So when the application is submitted, so we look at the data, there should be comprehensive data with respect to the quality, non-clinical, from the safety and efficacy. And when we have this comprehensive data, we can make assessment of the benefit and risk. And if this ratio is positive, then the marketing authorization can be granted. In case of ATMP medicinal product, it always many times happens that the marketing authorization holder is obliged also to do some post-marketing efficacy and safety studies. This is mainly due to the fact that ATMPs are very complex molecules, so there should be still some additional data to be generated, collected on the heterogeneous study population. So this is what usually happens. But looking at that, let's ask, are there any opportunities within this regulatory framework that in some certain circumstances, the patients can get promising medicines earlier? Yes. So the answer is yes. So within the regulatory framework, there are some regulatory opportunities which are mainly categorized between regulatory tools. Those have legal provisions that are foreseen in the legislations. On the other side, there are also several supporting activities that are run or by European Medicine Agency or by national competent authorities. So regarding the supporting activities, I think it was already mentioned today, it is very important that developers get in contact with the regulator very early in the development, mainly because to avoid any development failures, to actually plan the development of the product in a way that in the end, the success of the marketing authorization would be much higher in the end. But I would like to give some insight also in these regulatory tools. So so for these regulatory tools, what are the main drivers, criteria that can be used for early patient access? So different regulatory tools have different criteria to be eligible for that, but in principle, this should address unmet medical need, or it should be major public health interest, or the medicine should be important therapeutic innovation, or the medicine should address seriously debilitating or life-threatening disease, or if it's orphan medicine, etc. So if we look at the conditional marketing authorization, it is not specific for ATMPs, but it's also applicable to ATMPs. For those, the authorization can be given before the comprehensive data are available. So in order to address unmet medical need, but this is possible if the benefits of the early access would outweigh the risk due to the limited information that we have about the product. In this way, some medicines can be authorized several years earlier. But it is, the main criteria is also that we know that the additional data that will be generated, mainly the clinical data that still needs to be generated, that this will be possible to provide them in certain period of time. Another criteria is also that it addresses unmet medical needs, and this should be for the debilitating or life-threatening disease. And yeah, as I said, it should be very likely that the data will be provided. The conditional marketing authorization, it's first valid for 1 year, then the marketing authorization holder gets specific obligation that needs to be fulfilled in certain period of time. So after 1 year, this conditional marketing authorization is extended. And when all data are collected, then again, the benefit balance is weighted, and if it's still positive, confirming to be positive, then after several years, or 3, 4, 5, the conditional marketing authorization can be converted to standard marketing authorization. Another regulatory tool, which also could a little bit speed up the access of the promising medicinal product, it's the accelerated assessment. As I have shown before, even the assessment process is not so quick. It can take a year or 2, depending how much issues are there. But in principle, when we look at the standard procedure, it takes assessment 210 days. These are only the active days of assessment. You have to know when the first list of questions comes, there is a clock stop, so the applicant has time to respond to all these questions or maybe do some additional studies. And when it comes back, it's a second round and it takes another 60 days of assessment, etc. But for the accelerate procedure, particularly for the ATMPs, this timeline of assessment is shortened. It's about 250 days of active assessment days. But for this accelerate assessment, it goes very much in line with the supporting activities that are available to developers, meaning that the data should be quite mature enough when the submission is, when the application is submitted, which means that the developer should be always, all the time in contact with the regulator. So being or having scientific advices, several scientific advices, or being part of this PRIME program, which means that the regulators are involved in this drug development pipeline from the beginning of the development, so that the development is planned in a way that as much as possible fulfills the regulatory requirements. Still, during the assessment, if it turns out that there are still several issues coming up, then this accelerated assessment can be converted to standard assessment. So looking at this scheme, I just want to show you, so these are all the products, ATMPs, that were approved in Europe since 2009 to 2023. Some of them are not available on the market anymore, not because of the reason of efficacy or safety, but mainly because of the commercial reasons. So the marketing authorization holder decided to not market the product. But what I wanted to show you mainly is that in the last years, the number of products that are authorized, ATMP products, the number is increasing. And I have to say that many of those products were part of these accelerated regulatory tools. So, and also many of those products were part of scientific advices before the application submission, or were part of PRIME program. So all these regulatory tools were used so that the medicines could get earlier to the market. So all what I was saying up to now was mainly based to this centralized procedure, but there are still some alternative regulatory pathways that are in the remit of national competent authorities. And these alternative regulatory pathways also have legal provisions that are foreseen in the legislation. So one of them is non-routine preparation of ATMPs. It's specific for the ATMPs, and with other words, it is called hospital exemption. Exemption mainly because it is meant to be used custom-made ATMP that is prepared from time to time in case that there is no ATMP marketed in Europe. So the product should be used for the individual patients in a hospital under the responsibility of the medical doctor who prescribes the medicine. And the national competent authorities approves the preparation of such ATMPs, but still this hospital exemption needs to fulfill criteria regarding the quality, traceability, pharmacovigilance, which are equivalent to those required for the marketing authorization. The important thing is also that for this non-routine preparation of ATMPs, they should be prepared within the member state where this medicine is used. Another alternative regulatory pathway, which is not specific for ATMPs, but it's still a way how to accelerate access. It's a compassionate use program. It's used for the treatment option that allows the use of new innovative medicines that are not still authorized by European Commission. The main idea is that the medicines that are used within this compassionate use program should be already in the late phase of development or should be already in the process of obtaining marketing authorization application. One of the criteria is that it should address therapeutic innovation, and it should also address to be used for patients with chronic or seriously debilitating disease that cannot be treated with other drugs that are approved. And also, as usually with regard to benefit risk, although we do not have all information about this product, there should be a clear sign that there will be a potential benefit from the treatment when used within the compassionate use program. I would also add some other options, which I have to say are not so much frequent in relation to ATMPs, but would just like to let you know that there are some other regulatory pathways. One of them is a treatment on a named patient basis. Actually, here is again that the medical doctor decides for use of new drug, new medicine, which is obtained from the manufacturer directly. But it's actually very much dependent how this regulatory pathway is regulated within each member state. For example, in Slovenia, this named patient basis can be only applicable for medicines which are authorized outside the Europe. So it should at least have authorization in Canada or USA or Japan, but every country has its own legal decision on this pathway. There are also options of expanded access programs. Not necessarily every country has this option, depending what is in the legislation, but it's meant to be for the medicines which are already in the clinical trials. And if some patients have very much benefit from the treatment during the clinical trial, which will end at a certain point. And if medical doctor thinks that some patient would require to continue with this medicine, this is one of the options. And now I would also like to touch upon clinical trials. There are quite a lot of clinical trials also in Europe with ATMPs. And I have to say that clinical trials, when we talk about development of new drug or new medicine, we need to think about clinical trials because this is the most reliable way to get information about the efficacy and safety of the novel medicinal product. In this respect, also, the patient should always be considered for being included in the clinical trials before being offered, before mentioned hospital exemption or compassionate use program. The main reason is that clinical trials, regulation of clinical trials is designed in a way that patients that will be included in clinical trial will have minimal risk when exposed to this novel medicinal product or investigational medicinal product. And at the same time, they should benefit as much as possible from this investigational medicinal product based on the level of information that we have at that time about this product. But nevertheless, it should be a priority way for the patients to be included in clinical trials. So basically, to conclude on these regulation aspects for the use of ATMPs, for all these ATMPs, the pharmaceutical legislation applies only if it's differently written in the regulation for ATMPs. No matter whether it's a marketing authorization application or whether it's a clinical trial application or whether it's a hospital exemption, it's a possibility of the risk-based approach because of the complexity of these medicines. Good manufacturing practice and good clinical practice always applies no matter which of those regulatory tools is chosen. And there is also needs to be the pharmacovigilance in place as well as risk management plan and traceability. And the last but not the least, no matter whether it's a marketing authorization or is a clinical trial application or it's a hospital exemption or other way of using the regulatory tool, the benefits should outweigh the risk for patients. So I hope I have given you some incentives that drug development, even for ATMPs, can be facilitated, and I really wish you a lot of success on that pathway.
Since after a very science-heavy session, half of you are asleep, and myself included, we will do a little bit of an exercise. I will invite all the speakers who spoke now, two are exempt, to join us at the round table, but we will do a little bit of a game. And we will also thank our sponsors because they enabled us to do this. So everybody will stand up, and then I will read out the sponsors. Whoever's name starts with the same letter as the sponsor's name can sit down. And then we will see, because there are really many, we will see how many A to Z letters we are missing in the room. So are you with me on that? Because we are thankful to the sponsors. Everybody wake up. Sorry, that's what I meant. Thank you. So we will start with the Children's Medical Research Institute. Everybody starting with the letter C, sit down. The second one is ALS E, letter E, sit down. Delavska hranilnica D, letter D, you can sit down. Oh, many standing still. Amgen is A, everybody with an A, sit down. Then we go for Czech Center of Phenogenomics, that's a C. Everybody with a C, sit down. We had a seat already. I will try to remember that. Novartis is an N. Everybody with an N. Then N is also National Institute of Chemistry. Done. Caritas with a K. All right. Then Sartorius with an S. S. Sit down. 3D Pharma Exchange. Anybody starting with a 3? No. All right. All right. Then Soglasnik with an S. We had that. Fundación Columbus. F. F. Then we go to CT, we had a C, Viral Gen, V, somebody with a V, sit down. Društvo za kulturo inkluzije, D, we had that already. CTNNB1, which I have to read out. I don't know which one it is, sorry. You will tell me later. Mestna općina Ljubljana, so that's the Ljubljana city, M. Or L also, we can choose L as well, Ljubljana. Then we have the Mestna kavarna, which is the city coffee shop. So it's an M again, University of Ljubljana. This is Faculty of Arts, U. Anybody with a U, sit down. Site, we had an S. Institute Forest, anybody with an I, sit down. Then Associação, CTNNB1, I don't know which country, A. A's can sit down. Grand Plaza, this hotel, G. We go to Swix Biopharma, we had an S. Medias, I, we had that, M. Then we have the University of Ljubljana, Faculty of Education. Liams, L, no. Again, CTNNB1, Connect and Cure. 3D we had again. Planet Glasbe, Planet of Music, P. Then I will read out faster now, and we will see who stands in the end. Robotrade, an R. Bormia Meta, B. DSHMS is a D. Ultragenics is a U. Then we have some are repeating. Neuromotion with an N. Robotrade with an R. Metoda Krakowska, M. Pržeskole Forest with a P. Slovenska Filantropija, S and F. We had that. Red Cross Slovenia with an R. Sanolabor, S. ALS, we had a few of those. Some of those are repeating. Nibalabs, I didn't tell that one. N. And perhaps I forgot some of the sponsors. Now tell us your names. Julia, you can sit down. Sorry? Once more. Taisia, where are you from? Thank you. Thank you for coming from Ukraine. We have somebody from Palestine here. Somebody works in Israel. So we are with you guys. What's your name? Jennifer. We didn't have a J? No. Juan, J. And we had you before? Javiera, thank you. Tom, we didn't have a T. Spiller, next time, sponsor with a T. Sorry? Tanya, we had you before. Thank you very much. So everybody, I applaud you. Thank you very much. So it's about connecting. It's about being also alert to information that is coming. Science requires like a monk's attitude. You sit down, you study. You had a little bit of that, okay? Now it will be a little bit more fun, and even more fun is coming after that. So I invite our speakers in this session to come up. I shall not go through names, you know who you were. So please, you're invited, and be ready for some non-scientific questions. And if anybody would really like to join them, like we had Ahmed before and Tanya, raise your hand. Would somebody like to join? No. Okay. There are enough anyway, so. All right. Sorry, I apologize. So you two, Spiller, they are invited or not? Please. I messed up. All right. So think about what your favorite book is, sport. You will tell me about which country has the most PhDs, tractors, and gene sequencing machines in the world. Those are the questions for you guys. So who goes with the book and tell me why you like the book? I will ask you.
Take the question of the book.
You will take the question. Do that. Stephen King, will you tell us why?
I like scary movies and something horrific. Yeah.
All right.
Yeah.
All right.
Yeah.
Is there a favorite one?
Billy Summers.
Sorry?
Billy Summers.
All right.
Yeah.
Thank you very much. Somebody else?
My favorite book is Hitchhiker's Guide to the Galaxy.
I actually met Douglas Adams. I spoke with somebody before. That was one occasion where when I shook his hand, I didn't want to wash it for some time.
Yeah, he died much too young.
Sorry. Yeah, so I had a discussion with somebody earlier today that sometimes these references are very interesting. And the reference here was that sometimes we're inventing new words. And Douglas Adams, if you know, wrote a book about words that haven't been invented yet. So he invented a whole book of new words. So we saw a lot of new words today. Why am I asking about the book? Because this is a very complicated and long story, and we are all writing this book of gene therapies, ATMPs, GLPs, GMPs, GCPs, compassionate use, and whatnot. So it's a genre. So it's not science fiction, and it's not what is a thriller or what Stephen King is something in between. Somebody else with a book? No? What about sport? You prefer? Oh, then tell us about the book.
But I have said I have many favorite books, so I cannot choose just
Give
one.
us one, too.
Oof. It's difficult. I mean,
It is. It is. Imagine how difficult it is for the audience.
So, for example, I really like the doctor. Well, I don't know how to say that in English. Probably it's the doctor, el médico, in Spanish, for example. But I really love science books and also history books. I mean, it's difficult to choose just one.
All right. All right. I will come to you. Why Douglas Adams? Hitchhiker's Guide.
Well, I think I applaud Douglas Adams to be able to make a coherent story out of all kind of, in the beginning, seeming random stuff.
Yeah.
Which is sometimes sort of like what science is. Trying to make some sense out of everything that's out there and what we're trying to do and try to focus everything to something that's meaningful.
So it's a large body of ideas that becomes coherent somehow. So it doesn't stay a chaos in his world, right?
In the end, yeah, somehow it comes together, yeah.
It comes together.
Yeah.
Does it relate anyhow? Because, you know, that was simply the impression before when we were hearing from the last presenter, Dr. Vidic, or it was already earlier on, that when you're doing a GMP or a GLP, you have to record each and every element. Is that in any way similar to you? Is there a comparison in terms of the thinking process or the attitude process? I'm not saying it's science, it's different, right?
I think it's a little bit different, but well, when you think of a GLP and GMP, one thing that science has often struggled with, I think, is that as always, you see a lot of results, a lot of papers being published, manuscripts written over the years. But when you try to recreate what the researchers did, it's very difficult to recreate it. And those quality systems, they are there to make sure that we can recreate what has been done before. And unfortunately, the only way to do that is to very meticulously put everything down that you have done and every change that you have made, which is a lot of work, but it is really necessary, and now it seems like a lot of work, but if you need to go back on something 5 or 6 or 8 years from now, you are very happy that someone did all the efforts to do that.
Thank you, Ruth. This is great because this is exactly what I was trying to get with the metaphor of the book, because you basically said where in science you are struggling in a way, where you have to come to the end. It's possible to come, but it's not guaranteed. So it's not that all this body of work is beyond critique. It has certain points where you say, come on, is this a good scientific article? Because also what we would not like to leave is an impression that we were sort of telling the parents, well, there was all the safety precautions, there was this and that. And if something happens, we don't take the responsibility. Of course, you don't want to take the responsibility, but we are all involved. So it's very important that the information goes here and there. In a way, we are all authors of what's coming up. Again, as stated before, it's not that we will be successful, but like in a sport that we will come to, it's the play that matters. Of course, with the children, we want to get the drug, but what we are doing now is the play. So, sports. I'll give you another chance. What about sports? No, to
you. Well, I really enjoyed football some years ago, but now I prefer more basket.
Basket?
Yeah.
All right.
It's more dynamic.
All right. In basketball, are you following the Slovenian guy who is now... we are 3-0 down to Boston Celtics. In Dallas Mavericks, Luka Doncic is playing. Slovenians are very proud. Are you following NBA? No? Are you playing basket?
No, not at all.
Not
No,
at all.
I'm just saying it, yeah.
All right. Thank you.
Thank you.
So what about you, Jan?
Wow. How sweet. Yeah. So now I have a really new sport activity. Actually, a week ago, I bought 20 ducks, which are supposed to kill slugs in my garden. And
slugs meaning a snail or?
Yeah.
20 ducks.
Yeah, and every evening when I return from my lab, I have to run with them, educate them, and teach them to sleep in a shelter.
Can you send us some photos? Because that's super interesting. 20 ducks.
Yeah.
All right.
These Indian runners. Which
yeah, no. But why not 5 or 3? Why 20?
Because there was a discount. And
all right.
And I have read a lot of snails in a garden. They destroyed watermelons. They destroyed suceta. So I had to do some really big answer.
But you are destroying mice in your lab, not snails.
Well.
All right. It's all for the greater good. Correct. That's true. Some other, would you like to tell us about the sports?
Well, the only sport I actually do is skiing, like downhill.
Me too. Me too.
Well, there we go. I heard you can ski here during the winter, which I...
Yeah, but Slovenians are very proud of Slovenia, but not of the skiing resorts generally. I go to ski to Austria, and many people go to Italy and similar. Where do you ski then?
Well, we ski either New Zealand. I live in Australia, so either Japan or New Zealand.
All right. So somebody else? Will you tell us about a book or some sport?
Yeah, I love climbing.
Climbing?
Yeah.
So what do you climb? What kind of obstacles?
I like better the rock climbing.
Yes.
But I also do bouldering.
What about alpine?
No. For the moment, just indoors.
So you know who the best female climber in the world is?
Jan Jagamert.
Yes, Slovenian? Yeah?
Yeah.
Yeah? Have you met her?
No.
All right. We are very proud of her.
Yeah, me too.
You are proud of her? That's
I
nice.
follow her a lot.
That's great. So for those who don't know, she's a gold medalist from the previous Olympic Games, poised to win again in this year. She basically won everything that was possible to win. So we love Janja as well. I myself, I will not be off the hook in that sense. I like to walk a lot. I do skiing. I do exercises in the morning, Tibetan exercises. I feel it's very important to de-stress myself. So when there is too much, I want to de-stress and I want to take a break. The other thing in the books, many books, I have a similar dilemma than you have, but the book I wanted to bring out still, or the author, is the Argentinian author Jorge Luis Borges. Does anybody know the author? A few of you do, yes, there were some hands. So he has not very long books, they are short. He's known for talking about labyrinths, but I'm sure in the popular culture, perhaps he's best known because Umberto Eco put him into the name of the rose. So the librarian, the blind librarian, is an homage to Jorge Luis Borges, not to go too long in that direction. So the question was, which country has most PhDs per capita, most tractors, you know, the agricultural tractors, and second most penetration of Illumina gene sequencing machines? It's not a trick question. It's a very direct question. Yes, some people know. You tell us. Yeah, you can. Well, let's leave this one. You will ask the question now. Do we have the mic? All right, you ask the question. I will repeat it. So, yeah, I will, because we don't have the mics here. So just say the question. So why don't you sleep on the airplane?
I don't sleep in a sitting position. I can't. Just never happened. That's it.
All right. How do you manage? I guess you fly a lot. And what happens then? You come to the hotel and you fall down?
Pretty much.
Pretty much. All right. Some other questions from the room. Would somebody like to know what the humane side of people who know so much science is? No? Further questions from me then? The country in question is Slovenia. So it has an absurd amount of PhDs. We have too many tractors, and we are crazy about buying gene sequencing machines and many other things. Slovenians have this peculiarity, but I think it's very interesting to see how Europe is different. You know, you travel 100 kilometers, you actually find a different culture of cheeses, wines, traditions, etc. And then you go further, sometimes even not 100 kilometers, everything changes again. I think Europe, that is often seen as very bureaucratic, from a few Americans that I spoke to here, it's such a bureaucracy, and yes, we have problems with bureaucracy as well, but the advantage that we have is there is a lot of diversity, and the diversity is also here in the room. So Spela, would you like to add something?
Yes. Does this work? Can you make it work?
Can you put the mic for Spela on?
Hello? Yes, okay. So I just want to say, so right now we have 15 minutes left. So we have all these experts here on the stage, and that's a really great opportunity. So if anyone has any questions, anything basically you need to know, now is the time.
All right.
And, the questions are only in English. So you can say in Spanish and then somebody else can translate it for you.
All right. So we woke you up a little bit. There are some things that are in your head that worry you. Please come forward with those questions. So we're trying to put the things on the lighter side a little bit. Would our panelists? Yeah, we have a question there, please.
Thank you. My question is not scientific related, but it's more about the timelines.
Yeah.
So today we saw the time frames for individual processes and steps, but just to guess a little bit the when, more or less, we will have this product, if everything goes well and as planned, when we will be able to have this product commercially available.
Yeah, so maybe I can
Please do.
add the first part, and then maybe Christina can add that up. So we are really trying to push for every single day and every single month to get as quickly as possible. Right now with Viral Gene, we have even tried to reduce and improve the timelines. But on current timelines, the product will be available to use in children via clinical phase, so clinical trial or other options that were presented today during the summer. So in July will be the product available. Is that correct, Christina? And then they need to complete the quality control, which also takes some time.
So this is July this year or next year?
Next year.
Next year, July next year. Yeah?
Yeah, as I mentioned, these are tentative timelines, and we have to agree together about all the activities that should be performed for that. As you have highlighted during the presentation, is really important to ensure the safety and the quality of the product, and for that we need to run several quality controls before releasing the drug for the clinical study. But yeah, the tentative timeline is by summer to have everything ready to be shipped to you. So I would like to add that we are running everything as fast as we can. So we are doing many activities in parallel, overlapping them in order to be able to deliver the product as soon as possible, and we are working together in these timelines.
All right. Yeah, you will have another question?
Hi, me again.
Javiera, correct?
Javiera, yes.
Yes.
I would like to, a law question. If you have the authorization here in Slovenia to do the test, can it be used in any child of the world? But here, or do we have to have our laws?
So in Slovenia, currently there is only one child, Urban. So we are doing this protocol to include children from basically anywhere in the world. The clinical protocol is not ready yet, so we don't know exact inclusion-exclusion criteria. And we're also thinking about an option of doing a clinical trial in parallel, for example, in Australia or possibly also in Spain. It all depends on the community and how well organized the community in specific countries. But in Slovenia, for sure, we will have children from all around the world.
All right. All right. Their questions are coming up. The lady here and then, yes. All right.
Hi, I have kind of a two-part question. One is, what is something that the families can do to be a supportive partner for you as scientists and researchers? And two, I'm sure none of you went into science thinking that you were going to work with patient organizations. But since you are now, and how things have progressed and changed along the way, what about these families and what they're doing has sort of infused or transformed the way you do your job at the bench and in the lab, and like, I don't know, changed your outlook or your integrity and just goals?
Thank you very much. I will give the question to you. I found it very illustrative, very clearly set. So I think you were talking to the patients very much in your presentation. Can you answer this question?
For me personally, I'm highly motivated when I see all of you. I know what I do in the lab, but I love to know for whom I do this. So for me, like, you and your children are the center of my work. So for me, this is super driving. And the other question was?
So how has this transformed your work? So one was, how can families contribute to what you're doing? So you explained about the relationship you have towards this. What was the change? Did you observe some change when you started working with the patients, patient organizations, and families?
Yeah, actually, what I said is more about the second.
Then I mixed it up.
Maybe
That
I mixed it up.
Perhaps it's late in the day, and
so so I'm mixing everything in my mind as well. So I don't know how to say it, but I think the families were the center, like from the beginning, because in these rare conditions, if you don't do anything, nothing happens, because the companies are not interested in this, because it is not profitable for them. So Spela has done this huge work, and all of you like building this community, raising money, like millions of dollars and euros you have gathered, and thanks to Spela and all of you, we are collaborating. I didn't know Dusko or the National Institute of Chemistry, and we are collaborating all together towards this gene therapy and other therapies. So I think you are the main characters here, and yes.
Thank you for saying that. The main characters in this story is you, parents. So what's your name, just to get you? Effie from Washington. Thank you for coming. Thank you very much. The question is with you now.
And I'm not sure who should answer, but when you do the clinical, when you do the mice studies, are the mice always like young mice, or are there some older mice? I'm just wondering if those studies mirror kind of, our kids are anywhere from months old to years old. I just wondered if there's any kind of parallel there.
So how do you attribute for age of the kid? Jan, will you take the question, or will you take the question, or both? Go first, Jan, and then we will go to.
So I can speak from some experience what we have with several actually very precise patient models we are working with. And what is very important to realize is that actually the mouse development is much, much faster than human. So basically from the birth to weaning, when you have actually independent individual which is not dependent on mother, it takes only 3 weeks in mouse. So you can imagine how all developmental processes are extremely fast in newly birthed animals, newly birthed pups, that basically in 3 or 4 days of their life, you see actually very rapid changes in the growth, in metabolism, also in the neurodevelopment. So this is also something what has to be considered when you are working with a preclinical model, and you, for example, want to have the intervention time for kids. Then you have, for example, only 1 or 2 days in a mouse model where you can do the stuff. And of course, if you do it too late, when the developmental process are already over, then the efficacy study may give a false negative results, because you basically miss the window of opportunity. And you can also look on it from the different sides, from the opposite, from the aging. And again, the human lives, let's say, average, I don't know if it's something like 70 years, I would say.
80.
Yeah. And with the mice, you have the maximum what they can reach in a lab. Some animals which I forgot in my colony management software lived like 2.5 years. And this is the maximum what you can get. And if you, for example, want to see the long-term effect of some therapy, for example, the AAV-based gene therapy, you have basically the window about 1 year, 1.5 years, what you can investigate it in a mouse model. But with the kids, we are not talking about the therapy for a year or 2 years. So this is something what might be bottleneck in analysis of the long-term effect of the therapy in a mouse model.
Thank you. You're from the States as well, right? Minneapolis, or not? No, sorry, I mixed up with the other family. I apologize.
I just want to add to it. It's also important to remember that the mice that we work with are inbreds, so all the mice are identical. So it's almost like having twins, but millions of them, versus every human is different. So mice are predictive, but really we have to kind of be open-minded. So in a way, we, just because mouse doesn't show something doesn't mean it's not going to happen in human. We're way more complicated and way more diverse. So
Thank you.
Hi. I have a question. I'm not sure if it's going to be able to be answered, but how many doses are being manufactured for the trial? In other words, about how many children do you foresee being chosen to participate in the trial?
Christina?
So I cannot answer to your question now because we manufacture a specific concentration, and it will depend on the productivity of this specific product, of CTNNB1 product, and also on the concentration that that CTNNB1 is requesting. So it's something we are working in, and in the following weeks we will let them know the number of milliliters and the number of vials that we are projecting, but we are working on that right now and we don't have the numbers yet. Sorry.
A guess even?
An
estimation?
I cannot give you an estimation right now.
That's
sorry.
okay. Thank you.
Thank
But,
you.
sorry, I want to add to that again. Sorry,
Yeah,
so-
please do, please do.
But whatever the number is, it's a defined number. It's, you know, let's say 20. It's still a number of doses. So I think to follow up on the question of lady there, it's once those doses get used up and it's a good result, there's going to be another manufacturing campaign required and another one. So with Andrea, we just submitted a grant in Australia to the government to try to get money for another clinical trial. But you know, we're hoping for success. If there is going to be success, there's actually going to be way more money required to keep it going. So what the families can help is just keep raising money.
All right. Can you introduce yourself?
Yes, I'm Laura. I'm from Canada as well.
Yes.
So my question kind of follows along this train in terms of once we hit next summer and it's clinical phase time, is there going to be still ongoing production of the medication, or does that pause and you wait to assess the data and then start again based on those preliminary results? And then part two to my question is, if your child is in that phase 1 of the clinical trial, do you have a timeline in terms of if we're coming to Slovenia for this trial, how long we would be here kind of undergoing probably lots of blood work and testing and physiologic measures, I would assume?
I mean, I can try to answer some of that. So the foundation paid for a campaign. Yeah, so the campaign will be completed. There will be testing being done, and then the doses will be released. The patients, children, we're going to start getting dosed, and there's usually a number of measurements that are being, you know, that's going to be part of the protocol. At some point, decision is going to be made that it looks promising, and that may be when there's still available doses. So if there is money, decision might be made to trigger another production, which again is going to take some time to, it's faster next time, I assume it's a bit faster, but it still takes time. Otherwise, usually you would wait, finish the trial, analyze all the data, and make decision, do you go to the next phase or not?
Yeah, and to answer your question about how much time do the children need to stay, the clinical protocol is not ready yet, but currently they're talking about 6 months. And it's really, really important. This is also something Julia can tell you about. You can prepare during the dinner, the preparation for the clinical trial, and also afterwards, the management after the clinical trial. So these are really, really critical things.
One more question.
I will translate.
Bueno, yo creo que una duda que tenemos muchos padres es cuando se habla de un tanque de 250 litros, es muchísima cantidad. Entonces, ¿por qué todo queda en unas pocas dosis útiles para los niños?
So we have seen that you are mentioning 250-liter tank, which is a huge amount of liquid. So what Esti is wondering is how come that then that is getting reduced to just a little few doses in volume for a few little children?
Yes, so when we are talking about this huge amount of liters, we are talking about the cell culture in which we are introducing this DNA, not about the AAVs. So if you remember from my presentation, these cells are like the engineers that will produce the AAVs, And then these AAVs has to be concentrated and purified along the process. So we are removing all the impurities that we can have in this soup of cell debris, cell proteins, and our AAVs. We have to get rid of all of this material and purify just the AAVs. So we take out all the impurities, and then what we have is a very concentrated product that will be delivered in vials, and that's why we are losing so much volume, because we are not talking about AAVs when we are talking about 250 liters. We are talking about the cells in which we are introducing this genetic material to generate the AAVs. So I don't know if I have answered your
If I can put it in simple words, that will be like a soup with little vermicelli, and you're fishing the vermicelli out of the soup.
Yes.
Yeah, I think it's very important to understand also that there will be many questions that will be coming up. Spela just said, please send questions to Spela. Of course, there are more people involved. It's important to hold these things together in a way, and the answers should be provided. Some answers can't be answered at this point because it's still a path that we have to learn for this disease. So it will take some time, but the outline is here. I think it's a very clear outline in comparison to some other diseases that we're talking. It's not clear at all how those will be treated. So it's a new territory. It's going into the dark. We will have to finish at this point. We'll be happy to take more questions afterwards, but please send them to Spela. I will happily be involved also in the future, our institute for sure. Thank you very much. Sorry for putting you on a little bit of a fun program, because I really wanted to introduce the next, he's not a speaker, he's an entertainer. Thank you very much for now. So become the audience now. And so Spela, if I may, I will introduce who the person is. Basically, I will use the term, the scientific term for these kinds of gatherings is symposium. So symposium comes from the Greek word that actually means a drinking party. I hope you have drunk a lot today, or that you will be having fun from now on. Sorry for the plug before. I do understand that there is a very serious issue behind in your head somewhere all the time. So I do acknowledge that. Now we have a special entertainer. He is somebody I met before together with his wife. He won a Golden Buzzer Award at the Britain's Got Talent a few years ago. So he responded to Spela's plead to come, and I spoke to him before, to his wife. They told me they've been married for 41 years. I don't believe them because they look 42 years old, both of them. There's his wife, and there is the guy who is known by his act, Wiggle Wine. I'm sure we will all enjoy this immensely. Thank you very much.
One, two. Hello, everybody. Hello. Thank you all for inviting me here. I'm very thrilled to be here and be a part of your movement and what's going on. So I'm very honored. And I'm now gonna lighten up the situation a little bit with a little bit of my Wiggle Wine feel good song. Okay? But before I even do that one, we're gonna level up the scene with a little Lionel Richie for you. All right, how y'all feeling, by the way? How you feeling? You feeling good? All right, let's change them. to feeling even better. Little Lionel Richie, please, huh? Come on. Woo! Woo! Da da. Whoa, whoa. Well, my friends, time has come. Raise the roof and have some fun. Throw away the work to be done. Let the music play. Everybody sing, everybody dance. Lose yourself in wild romance. We're going to forever. Come on and sing it along. We're going to forever. Forever. Come on and sing it along. Come on. See the rhythm rolling there. Let the music play. Feel it in your heart, feel it in your soul. Let the music take control. Come on, we're going to fly forever. Forever. Come on. Troubles away. Come join the podcast. See, no play. Tambolina Sentimonia. Way jumbo jumbo, way to party home we going. Oh, jumbo Tambolina Sentimonia. Yeah, jumbo jumbo. Well, everyone can jump in. It is a rip. Thank you. Thank you. Okay. Okay, everybody. See that in the mic? 1, 2, 1, 2. Okay, yeah. Okay. Hold. Hold. Now, just as I was out there, a couple of young ladies says, we know how to do wiggle wine. And we want to do the wiggle wine. So we could do with some participants. Anybody brave enough to come up here and join me in doing the wiggle wine? Oh, yes. That's what I'm talking about. Come on, man. Oh, yeah. Oh, yeah. He's taking a seat. Come on. Come on, ladies. Come on. We got some ladies. Come on. Come on. Come on. Let's do it. Demonstration. Let it go. Let it go. All right. So, hands up who have seen the wiggle wine dance. No, you've not seen the wiggle wine dance. We've got to show the wiggle wine dance. So it's all about the shoulders going like this and the waist doing like this. And you got to do both together. And then you can dress it up in your own style, So once you get the foundation, just do your thing. Yes, sir. Play it again. Here we go. This is a rumor. Come on, everybody. That's just because of the kids second man. I'm in the remix. You know how it goes. Come on. Pretty girl, you look sexy. Pretty girl, you look pretty. Pretty girl, you look sexy. Pretty girl, you look pretty. Wind up your body, girl, raise it high. Wiggle up your hips, them and take it to the sky. The music sweet like the food that you eat now. Come on. You look good, feel good, this is state of mind. Show off your body, girl, you know you look fine. Sweet soul music, no cleansing your soul. Your body is your temple, you never go home. Wiggle out and go back. Wiggle out and get look sexy. Wiggle out and your body. Wiggle out and get look pretty. Right. Wind up your body, girl, wine. Wind up your body, dropping low one time. Wind up your body, girl, wine. Wind up your body, girl. Again. Wind up your body, girl. Wind up your body, dropping low one time. Wind up your body, girl, wine. All the sexy ladies. Pretty girl, wiggle and wind your body. Wiggle and wind, girl, you look sexy. Pretty girl, wiggle and wind your body. Wiggle and wind, girl, you look pretty. Pretty girl, wiggle and wind your body. Wiggle and wind, girl, you look pretty. Hi, wiggle up your hips, never get to the sky. The music sweet like the food that you eat. You look good, feel good, this is get a mind. Show off your body, that you look fine. Sing soul music to cleanse the soul. Your body is the best, you never go. Wiggle out like your body. Wiggle out like you look sexy. Pretty girl, wiggle out like your body. Wiggle out and you look pretty. Pretty girl, wiggle out and your body. Wiggle out and you look sexy. Pretty girl, wiggle out and your body. Wiggle out and you look pretty. Pretty girl, wiggle out and your body. Wiggle out and you look sexy. Pretty girl, wiggle out and your body. Wiggle out and you look pretty. Okay, ladies and gentlemen, how we doing? We got time for one more? We got time for one more? We good? We got time for one more? Oh, yes, we got time for one. Yeah, man, I do what I can. Chapter 1 of Genesis. Only a man. Come on. I do what I can. Come here. I understand. Chapter 1 of Genesis, honey. I made a promise, darling. I got a bad heart, I want love. Y'all want me to wine and get down. So sexy, so sweet, that charming. Have mercy on me, my darling. I can hear my number calling. It's a blessing from above, you're receiving. I'm only a man. I do what I can. Don't you understand? Chapter 1 again, this is funny. I think the bubble is darling. Dancing burger, wind up your bumper. Jesus squeeze it, make your body feel it. Dancing burger, wind up your bumper. Jesus squeeze it, make your body feel it. I'm only a man, I do what I can. Won't you understand? Chapter 1 again, this is funny. I made a bubble feel darling. Every time that we jump, she wanna come stress me. Everywhere that we go, she wanna come test me. Dancing vulgar wine in your bumper, making me hot just matter. Teasing, squeezing, made me hotter, just raising up on no temperature. I'm only a man, I do what I can. Don't you understand? Chapter 1 again, this is funny. I made a promises, darling. At a hunch, make a take it to lunch, honey lunch. We can drink rum, run, get drunk. Stone jump, up, drop dead. We got new best friend. The key to be loving, girl, be well fed. Next about the love finish the street well. Come, girl, you look rosy. Wind up your body, girl, you're sexy. Next about the girl, let's have the party. All of the girls, then listen to the whoa. Whoa, wow, wow, whoa. Whoa, wow, wow, whoa. Baby. Oh, night. Oh, yeah. Baby. Oh, night. Baby. Sweet love you. I know what I can. I'm only a man. I do what I can. Won't you understand? Chapter 1 again, this is honey. I made a promise, darling. Yeah. Thank you so much, ladies and gentlemen. God bless you. Thanks for Spiller to invite us and all the people here. Wonderful, wonderful charity, wonderful movement. What you all did. Thank you so much. Thank you.
We are very close to finishing the official program of the conference today. But things are going on not just tomorrow and the day after that, but also today. We are organizing focus groups, so smaller groups where we discuss the disease, parenting, etc. We need 3 more volunteers for today. And we will need several more volunteers. Please talk to Spela. We just need 2 more for today. There is another one. Talk to Spela, please. Will you find Spela? And 1 more for today. There we have the gentleman. Thank you very much. We close this. Before you leave your translation devices on the tables. And this is how we finish. But I have to express gratitude, not just on my behalf, and not just on behalf of Spela, the organizing team, and everybody involved here, but I hope that the people grateful to you will be the ones who don't know that we together are creating something. I guess there are many people who were not here that will benefit from what we have been doing today, tomorrow, and the day after that. Let's keep these connections that are starting together. It's a long, long path. So I've known Spela quite for some time. I'm trying to help. Many people are trying to help. It's important to have this for a long time in the future. Perhaps we will show a way for other rare diseases as well. Thank you very much. And I'm touched as well. I see some emotions. Thank you very much. And again, nothing else, but thank you very much. Thank you.
Recording stopped.